just want to show you that all these different possibilities of types of interferences provide for a really wide variety of the types of research that people do here. And consequently, you're going to hear talks about a lot of different topics. And an additional angle that some people work on is how the tumor microenvironment interactions control the cancer or don't in some cases. So mostly my talk is about thanking everybody. This program doesn't happen on its own. It takes a lot of people to make the summer happen. And besides myself, the two other members of the CB education team who are sitting down here, Ines and Rim, were critical in doing the planning, running our sessions, and mentoring the students. So this really wouldn't have been possible without them. Thank you both. And then next up would be all the faculty who provide spots in their labs for the students to spend their time. And we have quite an array of faculty who are sort of pictured here. So you can see there's already a lot of people involved, but then many cases, the faculty leading the lab have a lot of people in their lab, and those are the people that work most directly with your students. So you have all kinds of other mentors that are interacting with the students on a daily basis. And these are most of them, with a few I didn't have pictures of. And then all the people in the Y part of thank you here, I'll show you some slides of. We took a field trip this year. It was a new thing. And we visited the Neuromuscular Research Lab, which is across the river. And it's a very interesting and exciting place. And as you can see, there are quite a few people who gave talks or showed the equipment that they have, an amazing amount of equipment there, to the students. It was, we spent 3 hours there, and all these people in one way or another were helping the students while we were there. And Brad Nindle in red is the director of the Neuromuscular Research Lab. We also had a speaker series, and these people noted here talked to the students about their career paths, some of which were straightforward and some of which were not, and also about their research. And then there was the people who volunteered for the admissions committee, some of who were faculty mentors this year and some who are faculty mentors from other years. And this is a Herculean job. Just if you note down there, we had 106 applicants that put us first and another 78 that put us second. And we give some consideration to most of those. So reading all of those and figuring out who we're going to accept is quite a big task. So I thank the rest of the admissions committee very much for helping me do this. Special thanks go to our fearless leader, David Boone, who oversees the whole academy, and Stephen Jones, our project manager. So we still didn't get group picture yet. So I hope that when we're done this morning, before we head out to lunch, we'll take some time to do a group picture. So what have your kids been doing all summer? You're kind of wondering, you ship them off and they come here and they spend the day and you're like, what the hell are they doing? Well, they do a lot of pipetting. There's Jason and Lorenzo. More pipetting. And we sometimes use big pipettes. There's Arav who's also pipetting. Learning about stuff. Looks like slide fixation. And then they learn to pipette sterilely. They're doing work in the culture hood with various cell lines. And you'll notice that most of them have spent some time working in one of the culture hoods. And that's actually that incubator behind Avery is where the cells go to live when they're growing and the kids aren't working with them in the culture hoods. So once you've cultured your cells, you need to look at them. So they spend a lot of time at microscopes. There's Anthony and Nancy. They use computers to visualize their science. Sometimes it's readout from microscopes. Sometimes they don't know what they're looking at. Sometimes the mentors don't know either. Or you, over there's a lot of computers in the lab. Most of our equipment's run by computers, so the students are playing with computers all over the place, and getting data and doing other things. You have to concentrate when you're in the lab and be focused. Anthony's at another piece of equipment that is able to image data. Looks like a gel reader. They spend a lot of time analyzing their data and preparing for talks. We made them talk every week, besides their big talk here. Do a lot of shaking, which usually shakes you at the same time. Lots of playing with chemicals. Here's loading a gel very carefully. Just want to emphasize that while they're working hard, being in the lab can be fun. See, they're enjoying themselves. Some of the time at any rate. Not sure about Sam there. You didn't give me one. And here are some of the students with some of their mentors. They do eat. We allow them to eat. You wouldn't believe it, but that's me peeking around the corner. So we did this field trip, and this is a lab that studies the science of fitness and sport and how the body responds to exercise. So they measure bone density, and they have this cool DEXA machine to do that. They also have one of the 26 in the country high-resolution peripheral quantitative CTs, which can look at bone density in very great detail and give 3D structure to the microarchitecture. And so, but this machine can only do the lower leg. You see that rider thing she has there? You have to sit in a chair and put your lower leg there, and then it slides into the machine. The students all wanted to know whether they had data from several football players. They kept asking about the various, but they weren't allowed to see that because they would have broken HIPAA rules. There are various machines to study the way muscles are working and strength of different parts of the body. So this fine fellow was educating them about this device. And then, yes, so Lorenzo volunteered to do this and see how strong his isometric strength was in his legs. Well, I guess it's at your arms. You lift the thing, but you're pressing down, your feet press down on those things on the bottom, which is actually measuring the tension. So he got a readout on that. They do oxygen intake of lung volume during all kinds of different exercises and conditions. I didn't even take pictures of everything they have there, but the other cool thing they do is they look at neurological stimulation of the brain to see how that impacts fitness responses or recovery. So this is just a composite of some of the group shots that I thought pulled most of them together, because I don't have we were missing 3 who couldn't come for one reason or another. So notes for we will be recording and we're Zooming. Our first 2 talks, the introducer should be on Zoom, hopefully. There will be a 15-minute break at about 10:30. You guys all know there's refreshments out there. Lunch is going to be down the hall and to the left in that open area. And then at 2 p.m., there's going to be a closing ceremony back in this room with the entire academy. And to remind you guys, when all the talks are done, it would be great to gather the kids down here and do some group shots. Parents might like to take those pictures for themselves as well. Okay. So what time is it? 8:40. Oh, right on the button there. All right. So let me escape from that. So I need to stop sharing. Okay. So Tim Burns is going to introduce Jason, who is our undergraduate, who's returning. This is his 3rd summer with us.
So it's my pleasure to introduce Jason Chang today. Obviously, this is his 3rd time around. 2 years ago, when he was a rising senior, he was actually in my lab, and the last year he's in a different lab, and he came back now after finishing his first year at Johns Hopkins. And he's now being funded by a Doris Duke Clinical Foundation award for this work, and it's just been amazing to see Jason grow over the last 3 years. He's really done remarkable stuff from the beginning, but it's just been quite rewarding to see him advance. And I'm quite excited that in a really short amount of time, he's made some significant progress towards one of our projects. So I will let Jason take it from here.
All right, thank you for the introduction, Dr. Burns. As you said, I'm Jason, and this year I work in the Burns lab, mainly focusing on a sector of lung cancer known as non-small cell lung cancer, and how the metabolic pathways regulates like enzymes involved in that. So for some introduction, it's important to know that lung cancer is one of the most prevalent forms of cancer, and actually is the leading cause of cancer deaths worldwide, accounting for about 1/3 of cancer deaths total. And actually has the highest instances of brain metastases to the brain, with about 40% of patients eventually developing brain mets. And it's a very poor survival rate, with the median survival being like around 6 months. So this is why it's very important to focus on this, as there's no like brain specific targeted therapy that exists. So there's no like targeted method of hitting the brain. So within non-small cell lung cancer, our lab focuses on the MET pathway, and MET itself is a protein. It's a receptor tyrosine kinase that actually binds with its ligand, the HGF, which is the hepatocyte growth factor, and it eventually mediates many downstream activities that are involved in cancer, like tumorigenesis. And this pathway is commonly upregulated in non-small cell lung cancer through many mechanisms, some of which I've shown here on the left. Yeah, let me get a laser. So the one that I'm focusing mainly on is amplification, but as you can see, there's also exon 14 skipping and point mutations, and also protein overexpression. Now, this is targetable using FDA-approved drugs known as MET tyrosine kinase inhibitors. The one I'll be using is comatinib, and it works right here. But around 40 to 60% of patients usually don't respond, or they eventually develop resistance, which is why it's important to find other methods of targeting MET as well. Now, downstream of MET, there's this very important EMT transcription factor, TWIST1, which drives the resistance of non-small cell lung cancer, which I talked about earlier. It's one of the main factors why patients get acquired resistance or even have de novo resistance. And TWIST1 is overexpressed in about 40% of patients or cases, and is associated with increased risk of metastases, a more aggressive tumor phenotype, and overall worsened patient prognosis. So this is why it's very important to focus on this pathway, because it's one of the key pathways that need to be targeted. And then next, as you all know, cancer is actually a metabolic disease that undergoes reprogramming for several reasons, one of which is to generate more resources to support tumor microenvironment. And actually it undergoes this process known as the Warburg effect, where it prefers to go through lactic fermentation instead of going through the TCA cycle, even in an oxygen-rich environment, which is very interesting because this also decreases the pH, which supports the tumor microenvironment in its processes, but also this results in an intake of glucose that's heavily increased. And this is important because so on the left here, we have a heat map of RNA sequence from patients who have brain metastases. And through pathway analysis, we found that those who had MET amplify, which is the pathway I was talking about earlier, had an increased association with glycolysis. Now, this is very interesting because whenever we took we further investigated this by taking cell lines from the same patient, or they're derived from the same patient, one of which was a metastatic line, the other was a primary tumor. If you see here, the 1993 is the metastatic line. Those all have increased metabolic activity, which is very interesting. So we're going to investigate this. And the whole reason why is, like I said, to find new ways of treating brain metastases, and this whole pathway and how it regulates glycolysis, which is the very first step of cellular respiration, is very poorly understood. So with that all being said, tying everything together, we hypothesized that the MET pathway is actually inducing key glycolytic enzymes to help drive non-small cell lung cancer whenever it's MET altered. So I have a diagram of cellular respiration here on the left with many key enzymes that I'll be investigating. The main technique I used was Western blotting, but I also did RT-PCR. The two that I'll go through each of these. The two that I had primary focus on were the two involved in glycolysis, which is converting glucose to pyruvate, which is what kickstarts all of cellular respiration. And the two enzymes are hexokinase 2 and PFKFB3. And there are also other parallel and distal pathways that I looked at, including lactic fermentation and TCA cycle. But like I said, the ones I highlighted in green, which are the first two, are the one I primarily looked at. So the first thing we did was we treated the H1993 metastatic cell line, which is also MET amplified, with MET TKIs to see if I had any alterations to the enzyme expression. And if you look here on the left, we don't really see any in most of the enzymes, but if you follow here to the right, whenever we retrieve comatinib, hexokinase actually seems to go down. Now this is verified because P27, which I will talk about later, should go up when we treat with comatinib. This is further supported with this heat map of RNA-seq, which is also 1993 cells. Whenever we treat with hexokinase, RNA expression actually decreases as a negative correlation. Now, what's very interesting is in these 1993 cells, what I talked about earlier were the vectors. So they had the same phenotype as a parental line. However, when we overexpress TWIST1, which is the key protein I talked about earlier, it actually seems to increase hexokinase expression, even in the presence of this MET inhibitor, which means I might be rescuing the expression of this downstream pathway, a downstream mediator target TWIST1. Now we see here that nothing else really changes. So for the rest of this project, I mainly focused on hexokinase. Now, this was a Western run by my mentor, Vinod, and he used H1993 crizotinib resistant cell lines, which is a MET inhibitor, so it's resistant to the MET inhibitors. And he actually saw that hexokinase overall was increased in the resistant cell line, which correlates with an increase in TWIST1 as well. Now, this slide, you don't see it very well. However, endogenous TWIST is very hard to detect, but you can see here very clearly that TWIST1 increases with hexokinase in the resistant cell lines. So then the next step was I wanted to see if this was on the RNA level. So we did RT-PCR with TWIST1 overexpressing 1993 cells, which are shown in red, and also the parental, well, it's not parental, but it's empty vector, which has the same phenotype as the parental. And we saw that in the TWIST1 overexpressing, the relative fold change or the relative expression of hexokinase RNA was heavily increased. And you can see here that even in the presence of comatinib, it seems to, although the standard deviation is a bit high, it seems to overall increase or even like seem to rescue the expression of hexokinase RNA. And you can see here with comatinib, you should go down in the empty vector line. Now, like I said earlier, P27 is one of the main mechanisms in which TWIST inhibits P27 that leads to resistance in patients. So this is RT-PCR done by one of my lab mates, Porva, and she found out TWIST1 doesn't really have any interaction with P27 at RNA level. So we hypothesized that was probably through the protein, so we did a Western with a cyclohexamide chase and analyzed the half-life of this. So just visually, you can see here on the top, this is the empty vector, when on the bottom is the TWIST1 overexpressing line. And just visually, you can see that P27 has an increased half-life compared to the TWIST1. So we can deduce on a primary level that P27's half-life is reduced by TWIST. Now, we wanted to see further through what mechanism, and we hypothesized that was through the proteasome. And MG132 is actually a proteasome inhibitor, which means it stops the degradation of protein. So whenever we add MG132, it seems that it's constant. The expression of P20 fluctuates, but it's relative. It doesn't go down in a very linear fashion. So from all of this, we can deduce that twist1 is suppressing the tumor. Oh, I should mention that P27 is a tumor suppressor, and it actually is decreased in expression by twist1 through twist1 inducing the proteasome degradation. So overall, twist1 leads to a decrease. Now, this graph isn't very good. I was rushed at making this. However, you can see just very roughly that in blue, we can see that the half-life of P27 is roughly 168 minutes, while in twist1 is 139 minutes. Whereas if we do a pulse trace with the proteasome inhibitor, MG132, it doesn't really seem to decrease. So this is just like visualizing the Westerns. And then the last thing I did was see if regulation of twist1 actually impacts the expression of hexakinase 2. Now, HGF, like I mentioned earlier, is the ligand for the MET pathway. And whenever it binds to MET, it mediates many downstream processes involved in tumorigenesis. Now, HGF is a hepatocyte growth factor. And whenever I initially ran this experiment, I put it in the presence of fetal bovine serum, which may have interfered with the effects of HGF. However, if you see here in the first two lanes, whenever there is no FBS present, meaning there are no naturally occurring HGF in the media, it seems to go up whenever HGF is added. Now, it's important that this is a MET wild type cell line, which means that twist1 is not expressed highly. So this is good because whenever we induce the expression of twist1, it seems to correlate with an increase in hexakinase. On the reverse, whenever we used Harmine, which is a twist1 inhibitor, the expression of hexakinase actually seems to go away significantly, which is which further supports our hypothesis. And overall, just from this Western or from both of these Westerns, we can deduce very roughly that twist1, whenever we increase or decrease the expression of twist1, the same thing would happen relatively with hexakinase. So overall, to summarize, I feel like this figure sums up my work nicely. So we have the MET pathway with the many mechanisms of MET dysregulation shown here, which all lead to twist1 overexpression. And twist1 overexpression, as I shown, induces the proteasomic degradation of P27, which is one of the mechanisms of why patients get resistance to the MET inhibitors. And it also seems to have twist1 seems to regulate hexakinase. We don't know if it's direct or indirect. However, that's one of the things we can investigate in the future. And overall, we saw that regulating twist1 expression seemed to have an effect on hexakinase in the same manner as well. So future directions involve using a combination of twist1 inhibitors, the MET inhibitors, as well as hexakinase inhibitors to see if there's any interaction at all, and to confirm whether or not twist1 is like a direct transcriptional regulator of hexakinase to see what type of regulation it has. So yeah, that's basically sums up all my work. And I would like to thank everyone who's made this opportunity possible, especially the people in the Burns lab and those at the cancer biology site. And overall, I'd like to thank all these foundations for supporting me as well. Here I have a picture of all my awesome lab mates and mentors. So yeah, thank you.
Great. Does anybody have any questions for Jason? You guys are in awe. He's okay. So we need to, I have a question for you.
Yeah.
Yes.
Didn't want to interrupt him.
Yeah, same thing.
Yeah.
Okay, so the next one up is talk 2. We have our introducer again.
You just want to have Tim go ahead and start introducing them?
Yeah, Tim, you there? Tim?
Yep, I'm here.
So if you want to go ahead and start introducing Levi.
Sure. It's really my great pleasure to introduce Levi, otherwise known as Leviticus McGraw. I don't think he generally goes by Levi. And despite all the pictures, he's not always in a hat. So this is Levi's second summer in the lab. He is going to be heading off. He just graduated at Woodland Hills. He's actually heading off to Penn in a few weeks. And this is with a scholarship from the Gates Foundation. And it's been great to have Levi in the summer, the last 2 summers. It's fantastic to see people come back and see how much they've grown and how much they've evolved over time. And Levi's really just hit the ground running this year, and it's really been fantastic to have in the lab. I'm looking forward to what the future may bring for him, but I think it's going to be very bright. If he ever decides to come back, he's always welcome.
Thank you, Dr. Rance. Thank you. All right. Oh, you're good. You're good. Thank you. Thank you. Right there. All right. So, hello, my name is Leviticus McGraw. I'm a rising freshman at University of Pennsylvania. And this summer I conducted research in the Burns lab, and my aim was targeting metabolic pathways in MET-driven non-small cell lung cancer to treat brain metastases. And so in the Burns lab, we focused on lung cancer because it is the second most prevalent form of cancer, not including skin cancers, and it is the most common cause of cancer death. Considering it is the second most common or prevalent form of cancer, it's scary to realize that it develops the greatest number, greatest number of brain metastases. And this is an even bigger problem considering that 40% of patients already have a metastatic disease at the time of diagnosis, and no brain-specific targeting therapies exist. And so, the MET pathway is often dysregulated within lung cancer. And so targeting this is targeting for treatment of brain metastases is often dysregulated in lung carcinomas, and the MET gene codes for a receptor kinase, a receptor in the tyrosine kinase family. And as Jason stated, the MET pathway can be dysregulated in various ways, ranging from point mutations, if I could use. Ranging from point mutations to fusions, and then to the ones that I focus on, exon 14 skipping and amplification. And so the metastatic one here is the MET amplification, which is shown to be metastatic in brain metastases. 16% of brain metastases are this MET amp. And this can be targeted using MET tyrosine kinase inhibitors. However, resistance is inevitable. And so we need to find new targeted therapies for MET-altered lung cancer and brain metastases. And so also, as Jason stated, cancer is in fact a metabolic disease. And a part of this, and unlike non-cancer cells, cancer likes to reprogram its metabolism in order to do the following, as listed on the board: to generate ATP, generate nucleotides and amino acids, to generate signaling molecules, or ROS, which are reactive oxygen species, and to inhibit the tumor microenvironment. And another major component of this is the Warburg effect, which is actually pretty confusing because there's not much known about it, except for the fact that the cells like to undergo lactic acid fermentation despite oxygen being present. And when oxygen is present, that usually causes the reverse to happen. So that would cause the reverse of lactic acid fermentation. And so, as you can see, per transcriptional analysis, we found that in primary lung adenocarcinoma and brain metastases, there's a clear difference in which genes are regulated. And we found that for pathway analysis, that within brain metastases, you see an upregulation of oxidative phosphorylation, which I will call OXPHOS, and glycolysis. And the upregulation of glycolysis is pretty unusual considering that most brain metastases are solely dependent upon OXPHOS. And so we did a transcriptional analysis of MET amplified and MET non-amplified lines. And you can also see a clear distinction between which genes are upregulated, which is the orange showing what is being regulated. And glycolysis was seen to be upregulated in this. And so this proves to us that MET drives glycolysis within non-small cell and MET and non-small cell lung cancer. And so in order to basically hammer this point, you can see these glycolytic proteins, specifically hexokinase, you can see that it has increased activity within the MET amplification line in comparison to the MET wild type. And lactate dehydrogenase, which is a proximal enzyme, which happens like directly after glycolysis, that also has an increased activity, as well as glutaminase. And so this higher activity reveals that glycolysis drives MET, as I said previously, MET amplification. And that was even further confirmed using a seahorse assay, which was done by Casey Cargill, who works in the Burns lab. And this shows higher ATP production and simulated glycolysis within the metastatic line in comparison to the wild type. And so with all that being said, I hypothesized that MET-altered non-small cell lung cancer will be uniquely sensitive to glycolytic inhibitors, and these inhibitors will be effective in MET tyrosine kinase in MET TKI-resistant cell lines. And so in order to do this, the method that I use is a CTG assay. And CTG assay is basically, it's a light-dependent assay, and it allows us to, or it quantifies the amount of ATP present and allows us to gauge the viability of the cells. And so for my experiment, I did two things. I did single therapy with H596 cell line, and these cell lines are right here listed. It shows you what the mutation or alteration is. I use the H596 cell line, the H1993 and H1993 crizotinib resistant. And I did that solely with metabolic inhibitors. After that, I decided to go on and do combination therapy within the H596 cell line, and I used MET TKI topoteninib. And I also added metabolic inhibitors to that, specifically glycolytic inhibitors 2DG and PFK, which both inhibit hexokinase 2 and PFK-FB3 respectively. And at the end of this, I compared IC50s, and the IC50 is the concentration of drug that kills 50% of cells on viability. And so just to give you a forewarning, I put key terms here because synergy, additive, and as antagonistic. This will come into play later in the presentation. And so first, in the MET exon 14 skipping mutant cells, H596, inhibiting glycolysis was actually effective. And as you can see, for all three of these, actually, the IC50 is lower at the later time point, 72 hours in comparison to 48 hours. And that shows a delayed effect. And in the cell line, H596, senescence has been hinted at before. Inhibiting downstream enzymes in this same cell line was also effective as well. As you can see, using metformin and V9302, G-boxin, and CB839, which all inhibit downstream, those are effective, and they show, especially G-boxin, this shows a delayed effect as well. And G-boxin is pretty aggressive, and from a clinical standpoint, you usually want to see like a plateau and then a later drop, so you know that the drug is more controllable. And that's what you see with sort of CB839 and V9302. And so on to the combination therapy, which was surprising because it was actually antagonistic. As you can see that all IC50s starting from 0 micromolars of tepotinib with both metabolic inhibitors, the IC50 starts to increase, meaning that with increasing concentrations of the MET TKI tepotinib, less cells are dying. And so the only outlier was 5 micromoles of tepotinib with PFK at 72 hours. But the main trend that you want to see is that this is antagonistic, meaning that both of the drugs technically inhibit one another, allowing for cells to be viable. And so going back to H1993, so in comparison to the wild type, the H1993 resistant line showed increased expression of hexokinase. And in the H1993 wild type, hexokinase goes down with capmatinib, which is a MET TKI. However, in the MET tyrosine kinase inhibitor resistant line, that's not the case. And so we believe that targeting glycolysis should be more effective in the resistant line. And so it actually was more effective, but it was also equally effective as well. What I mean is that per the red boxes, you'll see where it was actually more effective. As you can see, the IC50 for AT101 is 13.2, and down here is 24.3 micromoles. And so I'm using that one as just an example to show that this is where using glycolytic inhibitors in the MET TKI resistant cell line was actually more effective. Now, instances where it was actually equally effective, those aren't boxed at all. However, there was one instance that deferred from that, and that was using PFK 158. And as you can see that in the parent cell line, using glycolytic inhibitor PFK 158 is actually more effective. And so from our data, I can conclude that pharmacological inhibition of glycolysis in distal parallel metabolic pathways was actually effective, and inhibiting metabolic pathways did not show a higher efficacy. Meaning it was not more effective than inhibiting those distal or parallel pathways. Combination therapy was actually antagonistic, and inhibiting glycolysis was equally or more effective in MET TKI resistant cell lines than in the parental cells. And so, yeah, I just want to thank everyone that worked with me in the lab, my mentor, Dr. Burns, and Dr. Stabil, Dr. Kumar. I want to thank everyone in the cancer biology site and the University of Pittsburgh, because without them, this wouldn't be possible. So thank you. Yeah. Oh, I'm slow today.
Like that. Yes, please give the microphone back when you're done talking. Your mentor here? There he is. Come on down. Yes.
Exactly.
Okay. You can just hold this probably for an intro and then give it to Nancy.
Okay. Hi, hello, good morning, everyone. My name is Bashir Lawal. So I am in Dr. Wang's lab. So I'm the mentor of Nancy Wang. It's a pleasure to have Nancy in our lab. She is a very dedicated and hardworking student, and I wish you all the best in your future academic endeavor. All right.
Thank you. Oh.
I'm confused. Oh, okay. Thank you.
Thank you. Hi, as Bashir said, my name is Nancy, and my project is two parts. One part is computational, studying immunotherapy using kinase signatures, and the other part is studying high-grade serous carcinoma in the wet lab, specifically ovarian cancer. So let's get into it. So in immunotherapy, there is a need for patient stratification because only 5% of patients respond to immunotherapy in all cancers. And those who don't respond to immunotherapy may respond in stable, progressive, or hyperprogressive manners. So they might progress, their cancer may not progress, it might progress, or it might exponentially progress. And patients who do respond have excellent prognosis. You could probably say that they are cured because the immune cells, they recognize the cancer and they have such a low chance of the cancer coming back. Compared to other therapies like chemotherapy, immunotherapy doesn't damage other cells. Chemotherapy targets non-cancer cells and cancer cells, while immunotherapy targets cancer cells. And it also has fewer side effects. Okay, so what I'm studying is called the KRAS, which is the kinase of immunotherapy resistant signature. So there was, given thousands of kinases, we had to analyze them and sort them by the strongest AURC, which is area under the RC curve, which is kind of like a strength of predictability. And given that, we sort about 10 kinases that are significant. And why are we studying kinases? It's because kinases aid in phosphorylation of the proteins, reproduction, and therefore it aids in reproduction, metabolism, and apoptosis, which are all vital functions of the cell. So in my project, I analyzed the KRAS for glioblastoma. And average length of survival for patients with glioblastomas is 8 months. And the 5-year survival rate is about 7%, which is very devastating. It's a devastating prognosis. And so that is why there needs to be stratification, so patients who do respond to immunotherapy can get that. So first, I want to talk about a little bit of how immunotherapy works. So in cells, there is an immune checkpoint, which is where PD-L1 binds to PD-1. And whenever they bind, it doesn't allow the T cell to activate. And so the T cell does not infiltrate the tumor, and the tumor can keep growing. However, whenever we use inhibitors, which are immunotherapy drugs, we can use which are called anti-PD-1 and PD-L1. When these anti-PD-1 and PD-L1 bind to the receptors, they can it activates the T cell because they inhibit the PD-1 and PD-L1 binding, and so the tumor can then infiltrate the a T cell can then infiltrate the tumor cell, and the tumor can be killed. Okay, so this first image here is the kinases, the 10 kinases with their AURC values. Given that, so this next graph is so those patients with higher levels or higher expression of certain kinases, of these kinases, when treated with immunotherapy, they don't respond because they have higher levels of these kinases. However, the lower level of kinases, these patients with lower expression of these kinases are sensitive. However, whenever there is a higher expression of these kinases, majority of patients are resistant to the immunotherapy. And oh yeah, the name of the immunotherapy is called nivolumab and pembrolizumab. And so the ARC here on the bottom graph confirms and validates our graph at the top, because the ARC, as I mentioned earlier, is the strength of the prediction. And a neutral prediction is about like a 0.5, but a 0.86 prediction is pretty strong. Next, we did a survival analysis of kinases given the patient data. And so the first graph here is a progressive-free survivals, meaning that survival that is stable. Their tumor is not progressing, it's a stable tumor. So the blue here is low kinase expression, while the red is high kinase expression. And so the high kinase expression you can see here has much lower probability of survival than the low, those who have a low kinase expression. So yeah, those who have a high level of kinases have a lower probability of survival than the low. And it's actually the probability of survival for the for the low level kinases is actually triple those who have high level expression of kinases. This next graph here is overall survival. So here it shows just progressive, stable, and hyperprogressive. So all these manners of cancer progressing. And it's about those who have low have about more than double the probability than those who have high. This next slide here shows the immune infiltrations. So immune cells, they can infiltrate the tumor, right? But you have to be sure that there's different types of immune cells, and they have different functions. There's three I want to point out, which is macrophage, the M0, M1, and M2, which are in the red boxes. So usually in this image, you can see that the macrophage 0 has a moderately strong positive correlation with the level of kinases. So whenever the kinase expression level is high, the macrophage M0 level is also high. But M0 is an undifferentiated macrophage, also known as a naive macrophage. And M0 can differentiate into two macrophages. M1, which inhibits proliferation, and in layman's terms, it is a good macrophage. And M2, which promotes proliferation, it's also known as a tumor-associated macrophage, and it's also known as a bad macrophage. But in the correlation analysis here, you can see that there's a negative correlation with these. So we predict that the macrophage did not differentiate because both are negatively correlated. Also, another thing I want to point out here is that cytotoxic immune cells are T cells. They are negatively correlated with the Chiral signature. So the higher the kinase levels, there is a negative correlation, meaning that there are low levels of these cytotoxic cells. So these T cells will not infiltrate the tumor. This image is kind of like a second order interaction image. We found that we identified cancer promoting proteins as functional partners of Chiral. So these kinases, there's proteins that are partners that help them like push out their functions. And here's another image of that in addition to my previous slide. So one image is functional enrichment and one is pathway. So one is looking at the function of all these chirests together in multiple kinases versus the pathway enrichment, which is different pathways it regulates. The OGC is the gene amount of kinases, gene count. So the amount of kinases that that may alter each. So like, for example, the nerve growth factor receptor binding, that is about like 4 kinases. And the strength of the enrichment is basically like the degree of enrichment. So our conclusion from the immunotherapy is that chirests can predict immunotherapy resistance and prognosis in glioblastoma patients. Another part of my project was the ovarian cancer, high-grade serous carcinoma ovarian cancer, and it is the most lethal and prevalent type of ovarian cancer. EphA3, which is efferent tyrosine kinase A3, drives cancer initiation, and it's known to drive cancer initiation, progression, and metastases. However, the therapeutic efficacy of EphA3 inhibitors in high-grade serous carcinoma has not been investigated, and that is what we are hoping to do in my project. Another drug we are using is the EGFR inhibitor. And EGFR is an epidermal growth factor receptor, and it aids in cell growth, and it's found in high levels in cancer. And the inhibitor, I didn't write that, but the inhibitor is predicted to decrease cell growth. So here's a breakdown of ovarian cancer, and majority of, as you can see, 90% is epithelial, and it can be two types, the low-grade serous carcinoma or the high-grade serous carcinoma, but the high-grade serous carcinoma is the most common. So what I did in my project is I treated my cancer cell line, which is DOA13, a known human ovarian cancer carcinoma cell line, with AWL2, which is the EphA3 inhibitor, and EGFR and an EGFR inhibitor as well. So in this next slide, I'm going to go over the cell viability assay. So we use the IC50 concentration to determine the concentration of inhibitors. And IC50 is what concentration is needed of the drug to inhibit a biological function by half. And so as you can see in the first image, we use about 34.5 nanomolars of the EGFR, but we use 64 micromolars of the AWL2. And then in this next slide, we did a colony formation assay, and EGFR and EphA3, both inhibitors, decrease colony formation ability of the DOV13 cell line. As you can see in the image, the cells have decreased, and as well as the graph, which has a better visual. Also the EGFR inhibitor is more effective than the EphA3 inhibitor. EGFR and EphA3 inhibitors do decrease the expression of their respective proteins. EphA3 inhibitor reduced the expression of EphA3 protein, and EGFR TK1 inhibitor inhibited the tyrosine kinase, and which then inhibited the EGFR expression of EGFR protein. And the EGFR inhibitor has a small effect on EphA3, as you can see in the Western blot. Okay, so in conclusion, we found that EphA3 and EGFR inhibitors decrease cell viability, colony formation ability, and protein expression. In the future, in immunotherapy, we hope to validate our preclinical findings by translating them into clinical cohorts. And in the ovarian cancer study, we hope to conduct further studies in vivo and in vitro. I would like to thank the Wang Lab, Dr. Xiaosheng Long and Dr. Busher, who introduced me. They helped me so much this summer, and even after having his baby, he still helps me so much. And Dr. Persa and Dr. Yolanda, who are in my lab, as well as the undergrads, Anusha and Jessica, as well as the CB education team that gave me so much advice about my presentation and improved my speaking, as well as the Hillman Academy coordinators. Thank you so much. Oh.
Good job. Thank you. So Franco is up next, and you're introducing. Share the microphone. Yeah. Nice to meet you. We've been emailing back and forth.
So good morning, everyone. So I'm Joe. I'm a current postdoc in Dr. Becken's lab, and I'd like to introduce Franco from Greensburg High School. Now, Franco has been a great addition to our team this past couple of weeks, and although he's known for his basketball skills, I've gotten to know him more for his hard work and motivation, and I'm excited for him to share more about what he's been working on. Thank you.
Thank you, Joe.
Just got a new pocket.
Good morning, everyone. My name is Franco Alvarez. I'm here from the Bacchus lab this summer working for the Hillman Cancer Center. Thank you all for coming today, and I'm excited to share with you a little bit about what I did over the summer. So my lab mainly focuses on novel therapeutics in treating cancer, mainly because many of the treatments that are commonly used today, such as chemotherapy and radiation therapy, are quite effective and commonly used for many different types of cancers. However, they don't really do a great job at just targeting cancer cells. They cause a lot of damage to normal healthy cells that is kind of part of the risk benefit that you take whenever you're treating cancer. You know, a lot of the times you want to just target the cancer cells, but you can't because these treatments don't target any kind of pathways or systems that are specific just to cancer cells. So one main difference that we look at in our lab is the cell cycle. Cancer cells are notorious for rapidly replicating, out of control, unregulated all the time. So we started here. There are major checkpoints throughout the cell cycle. There are three main ones, but one of the main ones we focus on is the G1S checkpoint. What these checkpoints do is kind of regulate the cell throughout the cycle and make sure that the cell is okay to replicate and make sure that everything looks good before the cell kind of irreversibly commits to replicating. Because if you have a cell with damaged DNA and you decide to split into two daughter cells, you're effectively starting the process of creating a tumor because you are kind of irreversibly committed to replication after you pass through the G1S checkpoint. So this DNA damage checkpoint has a targeted pathway that I was kind of studying over the summer. This is the CHEK1 pathway. It starts with DNA damage whenever there is a replication error. That leads to a compound called ATR. It's a kinase. And whenever ATR is activated, this pathway on the right is turned on, and it eventually deactivates cyclin B1 and CDK1, which are kind of the cyclins that pull the cell, that would pull the cell into the next phase of the cell cycle. So by deactivating these compounds, you're effectively putting the cell in what we call a rest, which is kind of like a pause in the cell cycle, so that the cell has time to repair its damaged DNA before it continues to split. What we wanted to do is actually change that. What we did was block ATR using an ATR inhibitor, and this led to a different pathway activation, which kind of bypasses, even if there is DNA damage, it kind of bypasses these steps and allows the cell to start replication and start the S phase of the cell cycle, even if it has damaged DNA. And what can happen during this process, if you limit ATR from doing its job, is deoxyuridine contamination in DNA. This is a process where a compound called deoxyuridine kind of replaces thymine in the nucleotide bases of the DNA. And enough of this compound can be toxic to cells. So this is kind of where we wanted to target just cancer cells into dying before normal cells. One problem that does occur, however, is if you just rely on ATRIs alone, thymidine, which is another compound, can actually re-replace deoxyuridine in the genomic DNA, which kind of wastes our time because we want to kill the cells. We don't want the cells to be rescued. So we kind of need another compound to act on top of an ATRI in order to suppress the actions of thymidine. This is called a thymidylate synthase inhibitor or a TS inhibitor. And we kind of want to prevent the rescue of the cells because we want these cancer cells to die. I kind of highlighted that's just those thymine bases that are replacing deoxyuridine in the DNA. So that's what a healthy strand of DNA would look like. But we want the cancer cells to look more like what's circled down there, where there's a lot of deoxyuridine contamination. So why are we studying this at all? Well, cancer cells replicate, like I said, at a very high speed and kind of out of control. So if we use these treatments on a batch of normal and healthy cells and also cancer cells, then theoretically the cancer cells would be affected faster because they are replicating faster, and the treatment would work first in the cancer cells as opposed to the normal healthy cells. So we're effectively targeting cancer cells, which is the goal of our lab, is to create novel therapeutics that just target cancer cells and leave more of the normal healthy cells alone. So my hypothesis for this summer is that a combined dosage of an ATRI and thymidylate synthase inhibitor will induce deoxyuridine contamination and cancer cell death. These are the methods that I use. I use two mice cell lines, CT26 and B16. They're well-established cell lines. I didn't have to do any touching of mice because I don't like that. But CT26 is a colon cancer cell line and B16 is a melanoma cancer cell line. We took these cell lines, we put them into tissue culture and well plates, and then began the process of putting them into 96 well plates so that we can do a combined dose therapy screening. This was my setup for CT26. You can see kind of I zoned out the outside cells because we don't really treat those since they often yield bad results. And then I split down the middle so that each plate would have two replicates of the same treatment so that our results were a little bit more broad. On the x-axis is pemetrexed, which is the thymidylate synthase inhibitor, and on the y-axis is AZD, which is our ATRI inhibitor. And then they have varying dosages from none to pretty toxic dosages, and DMSO is the control and effectively no treatment. For B16, one thing I wanted to point out is that we flipped the axes of the drugs just because we wanted to have an extra row for pemetrexed on the y-axis so that we could increase the dosage a little bit more just to see if there was any more of an effect and try to make our graphs look better. These plates sat in the incubator for about 72 hours after we treated them. And then once the treatments were done kind of running their course on the cells, we would start doing the cell titer glow assay, which is a cell viability luminescence assay. Levi kind of already mentioned the cell titer glow assay before, but it kind of measures the ATP in the cells, which is just, it's just correlated with how many cells lived, because the more ATP, the more cells that there would be. Therefore, it's called the cell viability assay. And then I used a software called CumBenefit to create these beautiful looking figures over here. So these are my two replicates of CT26 that I did over the summer. Each graph was a different plate. They look a little bit different just because I ended up flipping the drug axes, but they're effectively the same thing. Two things that I wanted to point out on these graphs and what to take note of is in the very top there, that is my control, that is no treatment. And you'll notice along the z-axis, it says % control. That is because every single treatment was compared in how many cells lived compared to the control. The control we kind of measured as being 100% cells living because there was no treatment. And then every other cell was every other well was compared to the original no treatment cells because, they should be dying. And then the blue is the synergy where we see the best effect of both drugs combining to kill the most amount of cells. Often whenever you're doing drug treatments, you could just overload a patient and give them the most toxic dosages, but that always has negative side effects. So the goal is to try to get the minimum amount of a drug that's going to have the most amount of an effect. So synergy screening is kind of how we analyze that effect and see which minimum amount of doses of drugs can we give a patient that will yield the best results. You can see this is the B16 results. They also look great. They look a little bit different from each other. That could have been due to many different factors of the experimentation, like maybe the cells didn't sit in the treatment long enough, but you can still see synergy is consistent in both graphs. So we do believe that combining an ATRI with a thiamidylate synthase inhibitor is an effective treatment for reducing cancer cell viability. Like I showed in the previous graph, the drug levels, dosages, and effects were not consistent between cell lines. So one of the major things we want to look at is why this is, and we kind of have to create custom dosages for each of these cell lines. So that's another thing to take into account looking forwards towards the future. And after this, we're looking to kind of test this on more cell lines to get more variability in our experimentation, and then hopefully eventually move into in vivo experimentation to observe these effects within the complex organism. And hopefully someday down the future, maybe cancer patients will be taking our treatments in the clinical side of things. So once again, I'd like to thank my mentor, Joseph, and my PI, Dr. Baccanis, for allowing me to work in their lab this summer, the site heads of cancer biology for putting this all together for us, and my fellow scholars for continuing to come into work every day, even though it was difficult at some times. And of course, Dr. Boone and Stephen for helping coordinate the entire program for us. So thank you for listening.
Anybody have any questions? So, as Sam's coming up here and his talk's getting, make believe that I am his mentor, Catherine Demonalis, because she's getting on a plane at the moment. So she gave me her words. So, she's a faculty member at the Kelman Cancer Center and a member of the Cancer Epidemiology and Prevention Program. Sam is a rising senior at Fox Chapel High School, and he was especially interested in learning more about cancer prevention, diagnosis, data analysis, and programming. This is my second year as a Hillman Academy mentor, and I was excited to have Sam work with me on our analyses examining the impact of COVID-19 pandemic on cancer diagnoses, treatment, and prognosis using data from the UPMC Network Cancer Registry. It includes cancer-related data on all patients diagnosed at UPMC physicians. While he did not expect to work on a data-focused project this summer, he said it was a valuable experience to see how we can use existing data to conduct analyses that can answer important scientific questions. And I know he was really fond of the trip, the research, neuromuscular research lab. So he focused on colon cancer, which he's going to tell you about here. Given that he noticed that few studies have examined the impact of pandemic on its diagnosis, and especially given it's the fourth most common cancer diagnosed, and that colon cancer screening was highly impacted by the pandemic. I am very impressed with what Sam was able to accomplish this summer, conducting literature reviews, learning our programming, and conducting preliminary analyses of data. It's my pleasure to introduce his presentation, and I hope that you enjoy his talk on changes in colorectal cancer diagnosis before and during COVID-19 pandemic in Western Pennsylvania. Okay, so stick that in a pocket and put that somewhere. Okay, there.
All right, all right. Thank you, Dr. Demonalis from a plane, and thank you for introducing me for her. My name is Sam Litwin. I worked with Dr. Demonalis, and I worked in my lab called Cubicle. It was not a fancy lab, but it was my lab. It's where I called home for 7 weeks. I focused on specifically changes in colorectal cancer diagnoses before and during the COVID-19 pandemic in Western Pennsylvania. So some background on COVID-19 and colorectal cancer. Hopefully we all know what COVID-19 is. We did kind of have to live it for 3 years of our life, actually still ongoing. The first case of COVID-19 was diagnosed in December 31st, 2019, and it was officially declared a pandemic on March 11th, 2020. The pandemic was just recently declared over on May 11th, 2023. And over this time span, there's been about 770 million cases of COVID-19 with 7 million deaths. Is this the, we focused on, for our data from 2020, we focused on 3 main waves of the pandemic. The first wave hit March, April, and May of 2020. It's when we all went into lockdown. Then there was a secondary small wave in July and August of 2020, which was the post-summer wave, but it was very minor. It was also a final wave that was November and December, which was the alpha wave. These were the 3 waves that we focused on in our data. We, for colorectal cancer, it's cancer that starts in the colon or the rectum. It is a 63% overall survival rate. It is the 4th most prevalent form of cancer, with being 10.7% of all cancer diagnoses each year. And screening for this cancer is prevention. Routine screenings detect cancer cells before symptoms are present, and so that it can be treated before it develops into anything serious. So what was our project? We have 3 overall objectives. We wanted to determine how colorectal cancer diagnoses changed during the pandemic compared to the prior years. We wanted to determine if there was a shift in the stage diagnosed at during the pandemic compared to the prior years. And we also wanted to determine if the median time to first treatment increased or decreased during these years. We observed multiple factors, but the main ones I'm going to talk about are the patient's age, race, gender, the stage of diagnoses, urban or rural residence, and the neighborhood deprivation index, or NDI for short. NDI goes from 1 to 5, 1 being people in the highest income and 5 being people in the lowest income. So what was our method? For data collection, we collected all invasive colorectal cancer diagnoses from January 2018 to December 2020 from the UPMC Network Cancer Registry. And we compared 2 patient groups. We compared 2018 and 19, our pre-pandemic group, to 2020, our pandemic group. We used 2 analysis tools. We used RStudio, which was used to interpret all the data, and we used join point regression to evaluate trends by weekly diagnosis with each year. We also used statistical tests. We used the Mann-Whitney U test and the Fisher exact test to obtain p-values from these tests. Each test is used to test the association between 2 variables and see if the change from one to another is statistically significant. In our case, we tested to see if there was association between what year you were diagnosed in and other variables such as stage of cancer and median time to first treatment. For the p-values, anything less than 0.05 means that there is an association between the variables. And if it's greater than 0.05, there's no association between the variables. So for our first results, we focused on the diagnoses by demographic. So in 2018 and 19, there were 1,682 colon cancer cases. 828 were in 2018 and 854 were in 2019. In 2020, there were only 752. This is a 9.2% decrease compared to 2018 and an 11.9% decrease compared to 2019. Then we focused on change in diagnoses by age, sex, race, and residence, so rural versus urban. And only one of these had a statistically significant change, and that's age. We focused on those over 65 versus those under 65. And we saw that the change there was statistically significant, as shown by the p-value of 0.039, which is less than 0.05. We see that those over 65, they go from 59.6% of the diagnoses to 61.3% of the diagnoses. So although it seems like a minor shift, it is statistically significant. For results, we also looked at stage at diagnoses. So we intended to see if there was any changes in stage when diagnosed, and we reviewed this by different factors. And the short explanation, there's no association. Overall, there was a little shift from people diagnosed at stage 1 versus people diagnosed at stage 4. There were generally less stage 1 diagnoses or early stage diagnoses compared to late stage diagnoses, but it wasn't statistically significant. This is probably due to the lack of data, because we're only Western Pennsylvania. There was only a small population. We also looked at the diagnoses by week. So we used join point to create the graph and the trend lines. So 2018 is this dark blue line right here. And over the course of the full year, there's only a 0.05% decrease, which isn't significant at all. And in 2019, the slightly above it, the light blue line, that only has a 0.1% decrease. But in 2020, we saw multiple increases and decreases. In the start of the year, which is this first little block right here, there was a 7.16% increase from January to March. But then once the pandemic hit and we went into lockdown, we saw a 17.41% decrease from March to April, which is this line, right, which is right in here. Then from April to September, there was a recovery of a 4.4% increase. And then finally, at the end of the year, we saw a 5.16% decrease from September through December when we saw the last two waves of the pandemic. We also compared late stage versus early stage diagnoses. So we were looking to see the ratio of late stage and early stage case diagnoses. And we did this by comparing how many late stage to early stage and taking late stage and dividing it by the early stage to get us our ratio. So in 2018, there was, which is this blue line, there was a ratio of about 4.1 or 4.8 late stage diagnoses to 1 early stage diagnoses. In 2019, it was 3.7 to 1. In 2020, that number jumped all the way to 6 to 1 late stage versus early stage diagnoses. And you can see the peaks here in April, May, and June. Now, this is because in March and April, when everyone was stuck in lockdown, they couldn't get their diagnoses. They couldn't go to the doctor to go and get screened. So this means that when they put it off and they got their diagnoses later and later, the stages increase. So we see it just after when the waves hit. And we also see this down here in August, September, and with 2021 data, we would have expected to see it in more January and February after we see this initial, after we saw the third wave. We also looked at the time to first treatment in days. So we compared 2018 and 19 to 2020. We have the N values, which is the number of diagnoses, and we have the median, which is the median time in days to treatment. So we compared it for all different sorts of demographics. Green here on the right means that the median went down, red means that the median went up, and blue means it stayed the same. Out of all of these demographics, only 4 of these we consider notable, and that's those under 65, females, those in NDI 1 and 2, and those that were diagnosed with stage 3 cancer. The decrease in median was considered statistically significant in these cases. So for example, in males, a drop from 20 to 18 isn't considered statistically significant because it could be due to the lack of cases, and that's such a small decrease in median compared to those over 65 who saw a decrease from 25 all the way down to 18 days. So in conclusion, the COVID-19 pandemic caused fewer colorectal cancer diagnoses in Western PA in 2020. The pandemic affected the timing of diagnoses, the stage at diagnosis, and the time to first treatment. Future research that we should conduct should include the 2021 data and examine racial and socioeconomic disparities and evaluate changes in cancer screening during the pandemic. I'd like to thank my mentor, Dr. Demanellis, who unfortunately can't be here, along with the contributors with this, Dr. Brenda Dygard and Emma Gray, and Sharon Winters and the UPMC Network Cancer Registry staff for getting us all this data, along with our 3 site heads, Dr. Galson, Dr. Lose, and Dr. Nassar, and Stephen Jones, Dr. Boone, and the rest of the Hillman Academy staff who helped us make this possible. Thank you.
So before you start, does anybody have any questions? No? So next up is Avery and Samantha. Yeah. Here, do you want to just hold it to introduce her and then you can help her? You might put this part in your pocket when you're so you don't have to hold it.
All right. Hi, I'm Sam. I'm a postdoc in Patty Opresco's lab. She couldn't be here today to introduce her, but I'm happy to introduce Avery, who's a senior at Upper St. Clair High School. She is currently in the process of applying to Penn State, Ohio State, where she intends to study biology. Her long-term career goals are to obtain a PhD in cancer biology. So her initial mentor is in the process of starting his own lab, and so she had to switch projects halfway through her program. So within 3 weeks, she had to learn an entirely new project, and I'm very impressed in what she has accomplished. She was incredibly fast learner in the lab, and she has an excellent hand on the bench. Her favorite aspect of the lab was working in the cell culture hood, and today she will talk about a drug that inhibits the enzyme telomerase, which in turn kills cancer cells. So take it away, Avery. So you can just.
Thank you, Sam, for the introduction. Before I start, I want to thank the Hillman Academy for giving me this opportunity. This summer, I spent investigating how chemotherapeutic thiopurines inhibit telomerase elongation of telomeres. Telomeres are located at the ends of chromosomes. They act like a cap holding and protecting the ends of telomeres. In this picture here, you can see there's a chromosome, and the blue on the ends are the telomeres. Telomeres are kind of like the ends of your shoelace caps, protecting your shoelace so it doesn't fray. When a cell divides, your telomere length shortens. When your telomeres get too short, your cell can't divide. When telomeres are absent, the cell tries to repair the damage, but if it can't be repaired, then the cell triggers apoptosis, which is cell death. So you can see in the picture here, as the cell divides each time, the blue, which is the telomere on the end of the chromosome, will shorten. Telomerase is an enzyme that is found in about 85% of cancer cells. Telomerase is able to elongate the telomeres, allowing the cell to keep dividing. As you can see in the first picture here, this is a non-cancerous cell. As time goes on, the telomere will shorten, and in cell division, you can see each time the cell divides, eventually it will lead to cell death. But in the cancer cells, as time goes on, the telomeres stay the same length since the telomerase is elongating them. You can see this in cell division. When the cell divides, it is able to keep dividing because of the telomerase elongating the telomeres, resulting in rapid division and a tumor. So there's no cell death. 6-thio-DG, or thio, is a telomerase inhibitor. Thio has been shown to compromise the telomere structure, leading to the uncapping of telomeres, resulting in rapid tumor cell death. 6-thio-DG is incorporated into DNA, which causes apoptosis in cancer cells, telomere shortening, and telomerase inhibition. 6-thio-DG in mice has shown to reduce tumor size in telomerase positive cancers such as melanoma, which is skin cancer, lung cancer, brain cancer, and colon cancer. HeLa cells are cervical cancer cells. They're the first immortal human cell line. The two cell lines we used were HeLa long telomeres, or HeLa LT, and HeLa very short telomeres, or HeLa VST. The first thing we did was a colony assay. Colony assay is a technique used for studying the effectiveness of specific agents on the survival of cells. Colony assays were done in two different cell lines, the HeLa VST and HeLa LT, to determine colony formation using different doses of the drug. As you can see in this picture here, this was cells treated with no drugs, and there are lots of colony formations here, versus the second one where the cells were treated with the drug and there's much less colony formation. We did this by first seeding our treated cells. Then we aspirated the media off of the cells, which is just nutrients for the cells. Then we washed the cells with PBS to clean the cells. We placed the plates on ice and fixed with methanol to preserve the cells. Then we aspirated the methanol. We added crystal violet. This stains the cells, allowing us to visualize them. And then we let that sit for 10 minutes, and we wash the cells with water, and we let that air dry overnight. Our results from this, the HeLa LT is in green and the HeLa VST is in purple. Over here, when our cells weren't treated with any of the drug, you can see there's a lot of colony formation in both different types of cells. Then we added 1 micromolar of the thio, and there's a little more colony formation in the HeLa LT cells than the HeLa VST cells. And when we added 2.5 micromolars you can see there's a significant difference between the two, and there's much more colony formation in the HeLa LT cells than the HeLa VST cells. The next thing we did was immunofluorescence. Immunofluorescence is able to identify and locate the biological molecules by using a fluorescence labeled antibody. Immunofluorescence was conducted to detect co-localization of the telomeres and DNA damage. To do this, we first seeded our treated cells on cover slips and let that sit overnight. We washed the cells with PBST to clean them, and we fixed the cells with PFA, which is formaldehyde. That preserves the cells. We rinsed with BSA, and then we added our primary antibodies, which was gamma H2AX, so we can see our DNA damage, TRFA, so we can see our telomeric sheltering protein, PNA, so we can see our telomeric probe for fish. After we added the primary antibodies, we covered them with parafilm, and we let that sit overnight in the fridge. The next day, we removed the parafilm and we washed the cells. And then we added our secondary antibodies, which were Go Anti-Mouse 647 for far red and Go Anti-Rabbit 594 for red. Then we washed the cells with PBST to clean them, and we mounted them on slides. Here were our results for the HeLa VST treated cells with thio for 14 days. In the first row is our untreated cells. This one will show our telomeres. So the green dots are our telomeres. You can see there's a lot of telomeres in our cells. The next one is DNA damage. You can see there's not a lot of red dots because there's not a lot of DNA damage taking place. And then the next two are the two merged together with the DAPI, so we can view our nucleotides. Our next row is our HeLa VTS cells treated with 0.1 micromolar of thio. You can see there's a lot less green dots in this, and this is because the telomeres were either damaged, shortened, or uncapped. In the next one, you can see there's a lot more red dots, meaning there's a lot more DNA damage happening since the thio was added. And then the next one are two merged together with the DAPI. Our conclusions are that VST cells are more sensitive to thio due to their immense reliability on telomerase. This is because the VST cells have short telomeres, and in order for them to continue to divide, they rely on the telomerase to elongate their telomeres, while the LT cells are not as sensitive to the thio because they do not depend on the telomerase as much as the VST cells. And this is because the LT cells already have long telomeres, so they're allowed to divide until they have short telomeres, and then they can rely on the telomerase. For future directions, we like to determine the impact of 6-thio-DG on non-cancerous cells and telomerase negative cancer cells. For acknowledgements, I'd like to thank the University of Pittsburgh, Hillman Cancer Center, the Hillman Academy, the CB education team, and the members of the Prescott Lab.
Good morning. My name is Daniela Muoio. I'm a postdoc in Elise Fucurel Lab, and I had the pleasure to mentor Lorenzo Aguilera, a student from Franklin Regional High School. This was Lorenzo's first experience in a lab, and he showed from the start an amazing ability to handle complex experiments and topics, especially the last one he's going to present today. Thanks to Lorenzo's curiosity and also confidence on the bench, we were able to reach all the goals that we set for this experience. And we want to stress the fact that the data I collect during these past few weeks will be used to write a grant that we plan to submit later this year. So thank you, Lorenzo. And he's gonna today present the role of part 2 in DNA replication stress for us. Thank you.
This part in a pocket somewhere, so you don't have to hold it.
Put this one.
Yeah, put it close.
Okay. Good morning. My name is Lorenzo Aguilera, and my mentors were Daniela Muoio and Elise Fucurel. And my project was the role of part 2 in DNA replication stress. So in order to first understand my project, you first need to understand part 2. So part 2, along with these other enzymes on the right, is an enzyme in the ADP ribosyl transferases family which catalyzes PARylation, which PARylation is the transfer of ADP ribose moieties on specific target proteins. The next thing we need to understand is DNA replication stress. So DNA replication stress is when the genome of a cell is exposed to stresses that can cause the replication forks to stall. So on the left here is a replication fork, and whenever there's a stress, it can stall. And if this DNA replication stress is not resolved, then DNA damage can occur, such as a double strand break. Now, part 2 is involved in the DNA damage response. So this leads me to my research question: does part 2 have a unique role in the response to replication stress? I hypothesize that part 2 promotes fork progression to prevent replication forks from stalling and undergoing replication stress. So over the past 7 weeks, I've conducted 3 experiments, the first being a Western blot, which allowed me to detect and identify specific protein. Second being an immunofluorescence, which allowed me to visualize proteins, which mark double strand breaks. And the last being metaphase spreads, which allowed me to analyze chromosomes to determine replication stress during metaphase. With my 3 experiments, I had 4 different cell lines. The first, HeLa-FAP, or wild type, which had no genetic change to it. Second, being PARP1 knockout, which removed PARP1 from the cell. Third, being PARP2 knockout, which removed PARP2 from the cell. And the last being PARP1/2 knockout, which removed both PARP1 and PARP2 from the cell. With the 4 different cell lines, I had 3 treatments. First, being an untreated. Second, being treated with 0.4 micromolars of afidecolene. And a third being treated with 2 micromolars of afidecolene. APH, or afidecolene, is a DNA polymerase inhibitor that slows down replication forks and triggers replication stress. So as I stated, my first experiment was a Western blot, which allowed me to detect and identify specific protein, which in my case was phospho-CHEK1. So to understand why I use phospho-CHEK1, we first need to understand the ATR-CHEK1 pathway. So whenever there's DNA damage in the cell, it activates ATR, which is the master regulator of the pathway. Now, ATR is a kinase, so that means that it can phosphorylate. So when ATR phosphorylates, it then activates CHEK1, which then triggers the phosphorylation of CHEK1, which is the protein that I'm specifically looking at, which can be followed by the Western blot to show me how much replication stress is induced. And later on the pathway, cell cycle arrest occurs, which is the stoppage of DNA replication. So these are the results of my Western blot. Now, if we look at the graph on the right, we see that the most replication stress occurred with the 2 micromolars, and specifically in the HeLa-FAP, or wild type cell line. This is unexpected than what we would see, because since PARP1 and PARP2 are used in DNA repair, we would expect that more replication stress would be induced in the cell lines that do not have PARP1 or PARP2. However, we can still conclude that afidecolene triggers the phosphorylation of CHEK1 in all cell lines, which determines that replication stress did occur. My second method was in immunofluorescence, which allowed me to visualize 2 proteins which marked double strand breaks. So 53BP1 and gamma H2AX were the 2 proteins that I used, and they're DNA double strand break markers because they are present in the DNA damage response. And they're necessary for the activation. They are necessary because when they're activated, they signal ATR and ATM kinases. So looking at my results, it looks like a lot. However, if we focus on the right, these are the nuclei that I imaged underneath the microscope. And we see that with the higher concentrations of afidecolene, we can notice that there are more foci, or the dots, as we go down. And we also see more in the HeLa-FAP, or wild type cell line. Looking at the graphs on the left, we see that the brown, which is the 0.4 micromolars of afidecolene, we see that there's more replication stress. Therefore, we can conclude that afidecolene triggers replication stress and causes an increase of double strand breaks. The last experiment that I conducted was metaphase spreads. So with this, okay. Sorry. So with metaphase spreads, so first we need to understand the cell cycle, which is G1, S phase, G2, and mitosis. So with replication stress usually, or not replication stress, DNA replication usually occurs in the S phase. However, whenever there is replication stress, it can cause a delay in DNA replication. And sometimes this DNA replication can catch up during the mitosis or the M phase. So for my experiment, I first treated my cells with afidcholine for 16 hours. Then I allowed the cells to recover for 6 hours, and I added CDK1 inhibitor, which essentially synchronizes the cells. Then I washed cells for an hour, and I added EDU plus coulsomid for 1 hour. The EDU is used because it is incorporated in the newly synthesized DNA. So whenever we look at the metaphase spreads, we can visualize where these events are happening in the metaphase. The coulsomid was used to arrest cells in metaphase. So after the cells are arrested in the metaphase, we then swirl the cells, fix them, and we spread them on the microscope slides to then be visualized underneath the microscope. On the left here is a metaphase spread from the PARP1 knockout. So this is essentially what we are seeing. So for my results for the metaphase spread, they were very good actually. So if we look in the middle in the zoomed in picture, we can see that there's 2 dots and there's 1 dot, foci. And these represent 2 different pathways. There's a single event pathway and a double event pathway. So looking at the graph, we can see that there's an increase in 0.4 micromolar, specifically in the PARP1 knockout. This basically means that more DNA synthesis is occurring in the PARP1 knockout, which then we can conclude that the lack of PARP1 forces the cells to perform DNA synthesis in mitosis, while cells without PARP2 cannot. So basically, PARP2 is what allows the DNA replication to occur in mitosis. Summarize, I conclude that cell signaling of replication stress is similar in cells expressing or lacking PARP1 and PARP2. However, the cell's response to stress is different. Cells lacking PARP1 catch up replication during mitosis, while cells lacking PARP2 harbor less synthesis events in mitosis. PARP2 may orchestrate the pathways driving DNA synthesis in mitosis. Unfortunately, I only had 7 weeks for this research project. So if I had additional time, I would like to conduct additional metaphase spreads and analyze those results and perform experiments in which the specific mechanism of PARP2 is shown. I would like to thank everyone in my lab for helping me on the bench and just understanding everything that was going on. I'd like to thank the cancer biology site education team for providing me necessary resources on how to give talks. And I would like to thank the University of Pittsburgh Cancer Institute, the Doris Duke Foundation, and the National Institute of Health. Thank you.
Very well done. So anybody want to pepper Lorenzo with some questions? Try to stump us? Okay. Well, there are no questions. You guys are released to walk around the bathroom, is go out and then left.
All right. Oh, it is on. Okay. Good morning, everyone. I have the pleasure of introducing R.F. Serpentini this morning. He is a rising senior at Moon Area High School. Like all of the talented students that we have here today, reading his CV is honestly very exhausting. So I'm just going to give you a couple of highlights. In his school, he's involved in things like the debate team, math league, varsity tennis, cross country. In the community, he volunteers with Carnegie Science Center, the North Hills Food Bank. He does a lot of work with his local library, and all while maintaining obviously a heavy course load full of AP classes and a stellar GPA. So I'm tired just talking about you. And at such a young age. Well, let's not talk about what my CV looked like when I was in high school. He is aspiring to go to med school and become a physician. I asked him at one point what he does to relax. And after looking at me a little confusingly, like he didn't even know what the word relax meant, he clarified that in his downtime, he likes to play tennis and watch reruns of the TV show Friends. So I think you're most like a Ross because you're a little nerdy, which is a compliment. Very passionate about science, probably going to be a
a doctor someday and will happily tell everyone that you are. He actually came to us last summer. So he reached out to our lab, the Nicole Chef's lab, and volunteered for a couple weeks and learned some PCR, collected some data that we actually used in a figure for an R01 application earlier this year. So as you can imagine, we were very enthusiastic to have him back this summer when he got into the Hillman Academy, and we're thankful for that opportunity. And so I got to work with him this summer for the past 6 or 7 weeks and mentor him, and I'm very excited to have him talk to you a little bit about the project that we've been working on.
Just go like right here. Is that fine? Okay. Oh.
Can you guys hear me? Okay. Thank you, Dr. McIlvert, for that wonderful introduction. Hi, everyone. My name is Serpentini. Like Dr. McIlvert said, I'm a rising senior at Mineria High School. And my project today is on neuronal PD-L1 expression and how that modulates baseline tissue immunity. So to start off, let's start off with a little background. So in a tumor microenvironment, we can see around 3 main parts. We have the cancer itself, we have immune cells, and sensory neurons. So let's look at immune cells here. Immune cells have a protein called PD-1. It's known as programmed death 1, which essentially what it's responsible for is regulating the amount of immune cells that are in a certain area. And cancer has an activator protein or an immune checkpoint protein called PD-L1, programmed death ligand 1. And essentially, like that cartoon shows you right there, it binds to the PD-1 receptor on these immune cells and smacks it in the head with a bat, or essentially shuts it down entirely, and allows the cancer to grow without any regulation. So it's been recently discovered that not only does the cancer in the tumor microenvironment have PD-L1, but the sensory neurons in the tumor microenvironment also express this PD-L1. And you can see on this graph here that if we look here in the DRG and the TG, which are ganglia or bundles of sensory neuron, this graph shows us that there is expression of PD-L1 in those ganglia. So that's the backbone of our experiment or of this project and why we're actually doing it. And the goal of this project is to analyze the role of sensory neuron PD-L1 and what that role is on baseline immunity. So in your how it affects your immune system without any disease or any injury. So our hypothesis is that as we lose PD-L1, we will see a loss of PD-L1 expression in pain-sensing sensory neurons will dysregulate baseline immunity. And what I'm saying by that is that a loss of PD-L1 expression in these pain-sensing sensory neurons, and as we lose it, we will see an increase of immune cells in those areas. So in order to test this, we use 2 different genetically modified mice. The first one is our knockout mouse, or our TRPV1-CRE PD-L1 flox mouse. So that's a mouthful, so let's break it down. So TRPV1 is a receptor for pain. So what this mouse is it essentially in all TRPV1 expressing neurons, we are knocking out PD-L1 from them. So that's our experimental mouse. And on our control mouse, if we want to think about taking out PD-L1 with scissors, the scissors or the flox is present in this mouse, but the CRE or the activator to take out or the hands needed to cut the PD-L1 is not present. So since we didn't make these mice ourselves, and these were gifted from another lab in Oakland, we have to confirm the knockout actually occurred. So to do this, we've been testing the trigeminal ganglia, or that bundle of neurons that innervates the head, as you can see there in like a 3-pronged motion. And so we'll be testing the PD-L1 expression in that in the knockout mice and in the control mice. And to actually measure the amount of immune cells and the effect that PD-L1 has on the immune system, we'll be analyzing 5 different tissue samples: spleen and lymph nodes, which are lymphatic tissue; pancreas, which is visceral tissue; and tongue, which is muscular tissue. So, and skin, somatic tissue. And we're analyzing all these 5 different tissues to get a wide variety of samples so that we can better understand the whole effect it has on the body. So in order to confirm the knockout of PD-L1, we run an experiment called PCR, which essentially measures relative gene expression. So how we do this is we extract RNA from the desired sample of choice, in this case, the trigeminal ganglia, and then we run this experiment PCR, and then we can confirm if PD-L1 has been knocked out. So what we hypothesize or we expect to see is a decrease in PD-L1 expression in sensory ganglia from the knockout mice compared to the control mice. Or we expect to see a decrease in PD-L1 in our knockout mice compared to our control.
And what we see is exactly that. We see a significant decrease in PD-L1 expression in the trigeminal ganglia from the knockout compared to the control. And you can see that on the graph as well. So what this tells us is that our knockout worked. We can confirm our genotype, and then that way this experiment, we can proceed with this experiment. So for the next part of our experiment, we'd actually be measuring the amount of immune cells that are present in our knockout mice compared to our control mice. And to do this, we essentially measure these various different types of immune cells that I've circled here. And we created a plate of all those different immune cells, and we run an experiment called flow cytometry. So how flow cytometry works is that we extract the tissue from the mice, and we dissociate it. We stain it for the different markers that each of these different immune cells express, and then we put it through a flow cytometer. And after we put it through the flow cytometer, we get results like this. So each of these individual blue dots are cells. And we're able, through a process called gating, able to quantify how many different immune cells or the various different types of cells that we're trying to measure and the percentage of them in our samples. So we can start off by gating for the amount of actual cells versus debris, and then go from there and analyze the different markers that each immune cell expresses, and then see how much immune cells are actually present in our sample. So after we do that, we get results, and we can get the results, and we'll go over those now. So to start off, let's look at the lymph node. I've highlighted the T cells here because the T cells are, number one, the most studied when it comes to the PD-1 and PD-L1 pathway. And also, we most associate those T cells with fighting cancer itself. And what we see in the lymph node, in the T cell expression of in these control compared to the knockout, is a decrease in PD-L1 expression from our control to our knockout. So let's revisit our hypothesis really quickly. So we expected to see that as we knock out PD-L1, we'll see an increase of immune cells. And we're seeing the opposite of that hypothesis in the lymph nodes. And we also see that same opposite effect in the spleen as well. So what does this tell us? This tells us that as we lose PD-L1, we also see a loss of these different T cell subtypes in lymphatic tissue. So this poses two main questions. One, are the T cells just not there? Or did they leave the lymphatic organs and infiltrate the tissue? So we can answer this by actually looking at the tissue itself. So let's look at the pancreas now. So when we look at the pancreas, we also see that decrease of the T cell populations. So that tells us that a loss of neuronal PD-L1 results in a loss of these different T cell subtypes in pancreatic tissue or visceral tissue. And then, however, let's look at skin. In skin, we see the opposite of the results we've been seeing before, an increase of PD-L1 expression in our knockout compared to our control, not just in T cell populations, but in all immune cell populations that we're studying in this project. And in tongue, we see very minimal results, if any. You can look here on the T cells, there's very minimal change at all. Cytotoxic T cells have a slight increase and helper T cells a slight decrease, but there isn't enough to determine a significant change. So in skin, we can see that as a loss of neuronal PD-L1 occurs, we see an increase of T cell subtypes in somatic tissue. And in tongue, we see no real definitive change. There might be a slight increase here or there, but no real definitive change in these T cell subtypes. So let's summarize our results. The lymph, spleen, and pancreas, we see a decrease in the T cell subpopulations. In the skin, we see an increase, not just in T cell subpopulations, but in all immune cell populations in general. In tongue, we see no real change. We see maybe a slight increase or decrease here or there, but not a definitive increase or decrease to say that there is a change, a significant change. So our overall conclusion that we can say is that PD-L1 on sensory neurons may regulate immune cells in a tissue-specific manner. So what that means is that the PD-L1 present on these sensory neurons may regulate these immune cells, but it will vary from tissue to tissue. So it's not tissue-specific. So where do we go from here? To start off, we have to further confirm that the PD-L1 was actually knocked out of these mice. So I talked about PCR and how that measures relative gene expression. Well, the next experiment we would do to do that is called immunohistochemistry, which essentially measures protein expression. And we would expect to see a decrease in the PD-L1 expression in our knockout compared to our control, similar to what we expected to see in PCR. And immunohistochemistry looks something like that. And in flow cytometry, what we'd expect to see is our future direction, sorry, would be to introduce cancer into the tumor microenvironment. That way we can truly get a gauge of how PD-L1 is affecting the immune system's ability to fight the cancer itself, not just on baseline immunity. So to wrap this up, I want to say thank you to the UPMC Hillman Cancer Center Academy for giving me this wonderful opportunity this summer. I also want to say thank you to all my lab members, specifically Dr. Sheff and Dr. McIlvern, for taking me under their wing and introducing me to this project and helping me learn and grow through these past 7 weeks, and everyone else in the lab as well that has helped me in tremendous different ways. And then finally, I want to say thank you to the cancer biology program leaders, Dr. Gulson, Dr. Losa, and Dr. Nasser. And finally, the overall program coordinators, Dr. David Boone and Stephen Jones. Thank you.
Patients. Can everybody hear me? Good? Yes, thank you. I'm Megan Atherton, and I'm a lab manager and researcher with the Sheff Lab. And this summer, we had the distinct pleasure of mentoring our next speaker, Ms. Tien Legrand, during her summer experience here at the Hillman Academy. So Ms. Tien came to us from Oakland Catholic High School, where she graduated this May. She completed college-level coursework in calculus, biology, and argumentation, in addition to holding several different leadership positions. She was the executive vice board president of the Black Student Union. She also mentored fellow high school students in the LEAD and FIAT programs, in addition to holding different volunteer positions and performing other activities in the summer, such as research last year and this year. So it's no surprise that she earned her entry into CMU, where she'll be matriculating in just a few short weeks. She plans to study neuroscience with the eventual goal of pursuing an MD PhD program. But before we send her off to do all of those exciting things, we had the honor of hosting her during her research on CGRP, anti-tumor immunity, and tumor growth. And during her time here in the Sheff Lab, Tien has displayed not only the intelligence and work ethic that you can infer from her high school career, but also a distinct aptitude for the scientific methodologies, the analysis, and the various different kinds of benchtop techniques that you're going to hear her describe. And below all of that, I think even more difficult to teach than PCR and cell culture and those sorts of things, she showed a deep attentiveness, thoughtfulness, a willingness to ask good questions while still trying to figure things out on her own and seeking out guidance at the same time. She showed a great adaptability. We faced several technological and experimental challenges, which is not uncommon in research, of course. And she flew right through them with such a positive attitude and an energy every day, just coming into the lab with just a great amount of intellectual curiosity and real ownership of her project. Personally, it was just such a pleasure to mentor her and have her in the lab and just get to know her over these past couple of months. So without further ado, and without further embarrassing her, please welcome Ms. Tien Legrand here to talk about her work in the Sheff Lab on CGRP and anti-tumor immunity.
I set it here.
Okay, just don't pull it off. So
Don't pull it off.
you stay pretty steady.
Thank you, Mrs. Atherton. Hi, everyone. Good morning. As Mrs. Atherton mentioned, my name is Tien Legrand, and this summer I had the privilege of working in the Sheff Lab looking at CGRP and anti-tumor immunity. Okay, next slide.
Down here.
Yeah, no.
Oh, this arrow. Okay. There.
There we go. Thank you. So the Sheff Lab investigates how different treatments of head and neck squamous cell carcinoma can alleviate pain and slow the rate of cancer progression. Currently, head and neck cancer patients suffer from severe pain as well as a high recurrence rate. Treatment for this often includes immunotherapy to bolster anti-tumor immune response, but patients often have a poor response to this type of therapy in particular. So calcitonin gene-related peptide, or CGRP, is a sensory neurotransmitter released during cancer pain. And recent data suggests a relationship between CGRP release and tumor growth, which is what you're seeing in the figure here. And this is the basis for our global hypothesis that sensory neurotransmitter CGRP inhibits anti-tumor immunity during head and neck cancer progression. So my work this summer centered around finding the relationship between these three variables: CGRP release, tumor cells, and immune response. It could be that CGRP has a direct effect on tumor cells, which then dampens immune response, or perhaps that CGRP has an effect on the immune system directly, which then causes tumor growth, or perhaps even a combination of the two. And with this is our working hypothesis that CGRP, when released from sensory neurons in the tumor microenvironment, activates tumor cells directly, driving the release of immunosuppressive proteins such as transforming growth factor beta, or TGF-beta, and program death ligand 1, PDL1. So we first looked at this relationship in vivo, trying to find whether or not release of excess CGRP alters tumor growth and immune response. We did this using a capsaicin assay. Capsaicin is the active component of chili peppers. It's what gives them their spice, and clinically it's used to treat neuralgia. And when capsaicin stimulates sensory neurons, such as SHRP1, which Arav talked about, it drives the release of CGRP, which we believe to be immunosuppressive. The process for this assay was to inoculate the mice with cancer. We then split the mice into two different groups, our vehicle and test group. The vehicle group was treated with 0.1% ethanol as a water additive, and the test group was given 1 micromolar of capsaicin also added to water. We then measured tumor growth and water consumption weekly over a 3-week period before harvesting the tumors for flow cytometry. And given the recent data implicating CGRP with an immunomodulatory effect, we expected to see an increase in tumor growth. And here's our data from this assay. As you can see, over the testing period, mice treated with capsaicin had significantly larger tumor volume when compared to the vehicle group. This data is further supported by our flow cytometry results, which show a decrease in cytotoxic T cells, which are the active cancer-fighting cells, in the capsaicin group when compared to the vehicle group, and also an increase in anti-inflammatory macrophages, which signifies a dampened immune system, or immune response rather. And this lets us know that mice that received capsaicin had larger tumors with less cancer-killing immune cells, which goes back to our global hypothesis that CGRP is inhibiting anti-tumor immunity. There are some limitations to this assay. As I mentioned, we measured water consumption as a confounding factor to ensure that the mice were receiving capsaicin to drive the immune response, and also just to ensure that the mice weren't dehydrated altogether. And we are seeing a dissimilarity in water consumption between these two groups. And in addition to this, assay does not provide the precise mechanisms by which this effect takes place, although we do believe we are seeing an effect on immune response. Because it is an alive animal, we are not able to definitively say that CGRP is acting directly on tumor cells, which is then affecting immune response, or if CGRP is acting on immune response directly, which led us to do our first in vitro experiment, looking at whether or not CGRP stimulation changes cancer cell gene expression, specifically looking at genes of interest, TGF-beta, which blocks T cells from recognizing cancer, and PDL1, which makes the T cells dysfunctional and allows cancer to escape. The process for this were to treat the cancer cells with the various levels of CGRP before harvesting the cells, extracting the RNA, and running qPCR. And with this, we expected to see heightened relative expression of our genes of interest amongst CGRP groups in particular. However, our data looking at TGF-beta when compared to our housekeeper beta-actin, which serves as a common denominator to which gene expression is normalized, we're not seeing much change amongst any of the groups. And similarly, the same can be said for the PDL1 group. Again, there might be some variation, but we cannot see a definitive change in this, which lets us know that CGRP treatment in oral cancer cells did not induce significant changes in the relative expression of these genes of interest, PDL1 and TGF-beta in particular. It could change relative expression in other immunosuppressive genes, but we are not seeing that effect here. We then moved to our second in vitro experiment, looking at whether or not CGRP stimulation changes cancer cells' ability to migrate. we did this using a scratch or wound healing assay, in which our treatment groups were CGRP in a high and low dose, complete media, which is media in the presence of growth stimulants or food as our baseline control, vehicle, which is media diluted with water, and serum-free media, which is media in the absence of those growth stimulants as our negative control. And the process for this was to scratch and treat cells with CGRP before capturing images over a period of 24 hours and visibly watching that gap close. And with this, we expected to see an increase in wound healing ability amongst the CGRP groups. So the graph to your left is showing the percent closure of cancer cells amongst the treatment groups at 6 and 24 hours. At the 6 hour, you can see media and CGRP low are very comparable, whereas at the 24th hour, CGRP low had a heightened wound healing effect, reaching almost 80% closure when compared to media in pink at around 60%, and certainly serum-free in gray at just about 40%. And something I think is really cool about this assay is you can see the effect directly on the plate in the scratch itself. Looking at the 6 hour mark, you see not much of a change between complete media and CGRP low. And then at the 24 hour mark, you can see that CGRP low is significantly smaller than complete media and certainly serum-free as well. And this lets us know that low doses of CGRP produced an increase in wound healing ability. So in summary, in vivo, release of endogenous CGRP is associated with an increase in oral tumor growth, as well as a decrease in anti-tumor immune response. In vitro, CGRP did not alter the relative gene expression of TGF-beta and PD-L1 in particular, and also in vitro, CGRP produced a slight increase in the wound healing ability of oral cancer cells. In conclusion, these results show that CGRP release in the oral cavity can increase tumor growth and decrease anti-tumor immunity. However, judging by our in vitro data, this effect is unlikely to be caused by direct activation of tumor cells. And further direction should continue to investigate the effect of CGRP directly on immune cells. I'd like to give many thanks to my mentors, Mrs. Megan Atherton and Dr. Nicole Sheff, for their abundant support and guidance, as well as Hillman Academy, the University of Pittsburgh, members of the Sheff Lab, Nicole, Katie, Kishmar, Dr. McIlvred, for all their unwavering support as well. Site head, Dr. Galson, as well as Dr. Nassar and Dr. Loza. And I didn't list her here, but my mom as well. You four asked really hard questions, and I think it really helped prepare me for this day. So I thank you for that. And also program coordinators, Stephen Jones and Dr. Boone, for making this experience possible for myself and my cohort. And thank you all for listening.
Anybody want to ask any questions? Quiet group. Okay, how are we doing on time? Well, we're running slightly ahead, but we can start to mosey on up. So next up is Desiree, and John is going to introduce her. You can go slow for a minute or two so we don't end up too out of sync. That's it. Slow-mo, slow-mo.
Still 5 minutes. I'm not talking that much. Good morning. My name is John Schmitz, and myself and Tofik had the opportunity to mentor Desiree Panza. Got it right, I think. She's a rising senior from the Western School of Technology and Environmental Science in the Baltimore, Maryland area. I think Desiree, like most young teenagers, has found a way to cram more hours of the day into a 24-hour period while maintaining her 4.0 GPA, very focused in physics and math. And how she got in our lab, I have no idea. Participating in sports, many committees and groups that she belongs to, on top of volunteering in the hospital. So very busy. Most of the time you get these young students, they haven't been in the lab, they haven't been exposed to all these techniques. And it's that first day or two that makes or breaks their whole experience. You know, you put a pipette in their hand and they go, what do I do with this? I'm like, well, this is your tool. She picked it up right away. By day 2, we were off and running. Once we started working with the cancer cells, after week 1, she was essentially on her own. She was just, you know, coming to me for some guidance. The only thing that stopped her during her period was the cells. They decided not to grow very well. She would have had much more data, but nonetheless, she has quite a bit as it is. I expect great things out of Desiree in the future, and I look forward to seeing her progress.
Thank you.
Hello, everyone. I'm Desiree, and I come from Maryland. And this summer, I was able to work in the Awani Cocoa Lab with my mentor, Dr. John Smits. And my project was on drug combinations with alisertib on small cell lung cancer. Small cell lung cancer, or SCLC, accounts for 15% of all lung cancers. It's a highly aggressive cancer. It divides very quickly and can easily metastasize to the brain or liver. Small cell lung cancer is strongly associated with tobacco carcinogens, and people with a history of smoking are at high risk for the disease. Small cell lung cancer is more responsive to chemotherapy than other lung cancers. However, drug resistance always occurs after about a year. This is why it's very important to research chemotherapy agents that'll more effectively treat small cell. Alisertib is an Aurora A kinase inhibitor. It's Aurora A kinase is an enzyme that helps the cell complete mitosis. It's critical for the proper formation of the mitotic spindle. Alisertib inhibits this enzyme, causing G2M block, which means that the cell can't move on to the mitosis phase from the G2 phase of the cell cycle. Which leads to cell death. In early clinical trials, alisertib demonstrated activity against small cell lung cancer, but single drug treatments are rarely effective due to the heterogeneity of cancer. We hypothesize that combining alisertib with approved drug treatments will enhance inhibition of SCLC cell proliferation. To test this hypothesis, we first did an immunoblot assay with DMS114 cells, which is a human small cell lung cancer cell line. To do this assay, we first plated these DMS114 cells in a 6-well plate with varying concentrations of alisertib, one untreated, one 100 nanomolars, and one 1,000 nanomolars. And we exposed one set to the drug for 24 hours and the other set for 72. And then we harvested the cell lysate, harvested the cells and created the protein lysate. We loaded the lysate into the gel wells and did electrophoresis to separate the protein molecules by size. And then once we get the membrane, we incubate the membrane with antibodies and substrate, and then we expose the membranes in film to be able to see the proteins. One of the proteins we probed for was PARP, and cleaved PARP is a signal of apoptosis. At a normal exposure of the blot, there is no cleaved PARP shown, but for any of the concentrations of alisertib. But at an overexposure of the blot, there's some cleaved PARP shown for 0.1 micromolars and a little bit for 1 micromolar. This means that alisertib causes slight apoptosis, but it's not its main mechanism, and that it induces cell death through other pathways besides apoptosis. And tubulin here is just a loading control, and it just shows that we put all the lines are equal, which means we put the same amount of protein in every well. And we also probed for phosphohistone H3 as well as total histone H3 because we wanted to see alisertib's effect on the G2M phase. Increased phosphorylation of histone H3 on CRI10 demonstrates a G2M block, which is what alisertib is supposed to do to the cell. We also probed for phospho-aurora A and total aurora A because alisertib is supposed to inhibit the active form of this protein, which is p-aurora A here. But when probing for this protein, we see kind of an unexpected result where alisertib induces p-aurora A at 0.1 micromolars when it's supposed to inhibit it. And we think that this is because the basal level of expression, as in like the untreated alisertib on p-aurora A is already so low. And so when we add alisertib to the cells to inhibit this protein expression decreases a little bit, decreases a lot in the first hour, but then the cell starts panicking and increases protein activity, trying to overcome the alisertib, which causes the induction of protein at the 0.1 micromolar 24-hour point. But over time, alisertib suppresses this activity, and that's why there's like little to no p-aurora A at the 0.1 micromolar point at the 72-hour. This graph shows the cell cycle. So you can see the p histone H3 helps us figure out that alisertib causes a G2M block. So that means the cell can't move on from this cycle to go into mitosis. So we repeated the 24-hour immunoblot to see if we would see the same results that we got from the p histone H3. And we did, and that confirms our results. And we also probed for PTCTP because PTCTP is a target of PLK1. And PLK1 and Aurora A work together in the cell cycle. So alisertib, the effects of alisertib on Aurora A would also happen to PLK1. So we wanted to measure PTCTP to see if the induction at 0.1 that we saw for Aurora A would happen for PTCTP. And it does, which confirms the result that we saw on the last blot. We also did a cell proliferation assay in which we combined alisertib with the chemotherapy agents onto the DMS-114 cells that we plated and then 96 well plates. And then we exposed the cells to the chemotherapy agent for 72 hours. And then we added WST1 reagent that helps us measure cell viability. And we take the absorbances that we get from this assay and we put it into Compusen. And this is a software that helps us calculate the combination index values that will help us to see if these drugs work together well. So this software uses percent self-proliferation data from the WST1 assay. We can also calculate the IC50 of the chemotherapy agents, and this is the concentration of drug necessary to reduce the cell proliferation by 50%. For alisertib, the IC50 was 206.2 nanomolars. For SN38, which is the active form of irinotecan, the IC50 is 13.2 nanomolars. And for paclitaxel, the IC50 is 1.8 nanomolars. Based on the calculated IC50 values for each drug, the drugs were combined in non-constant ratios. So these are our results from the drug combinations with alisertib and paclitaxel. And numbers, combination index values greater than 1 means that the chemotherapy agents are antagonistic and they don't work together to help treat the cancer. And equal to 1, it's additive and it's more like 1 + 1 = 2. They don't help each other, but the effects don't go against each other. And if it's synergistic, then that means the drugs are working together in treating small cell lung cancer. And for paclitaxel, we see a lot more synergy, with the most synergy being at 300 nanomolars of alisertib, and it's most antagonistic at higher concentrations of alisertib. For drug combinations with irinotecan or SN38, the active version, there was way more, the combination index values are greater than 1, which means that it was more antagonistic than synergistic and at best additive. So SN38 and alisertib don't work as well together as paclitaxel and alisertib. In conclusion, alisertib blocks cells in the G2M phase, and we observe cellular responses due to the G2M blockade, such as increased PORA and increased PTCP, which again is a target of PLK1. And alisertib in combination with paclitaxel was synergistic, while the combination with SN38 was at best additive. So future directions would be to determine the effect of drug combinations on protein expression and to evaluate the drug combinations in other cell lines of SCLC besides just DMS114, and to test the synergistic combinations in SCLC lung orthotopic animal models. Now I want to give a thank you to Stephen Jones and David Boone, who make this program possible. They're the program coordinators, and to Dr. Gaussen, Dr. Lose, and Dr. Nassar, who are the cancer biology site heads and who also make this whole site possible, and to Dr. Owoni Koko and Dr. Schmitz, who let me into their lab and taught me so much, and I appreciate them a lot. That's it, and thank you.
Go ahead, please.
Hi, everyone. Good morning. My name is Wen, and I'm a postdoc in Dr. S.J. Gao's lab. It is my honor today here to introduce Rami. Rami is going to start in PET this year, and he's going to major in neuroscience. And his career goal is to be a surgeon. He likes to play soccer, and also he collects music wells in his downtimes. But actually, I see him like very positive every day, and he's pretty enthusiastic on research. And we are really happy and appreciate that he joined our lab and worked during the summer. So during the summertime, he's mainly focused on the M60 modification, regulating the Kaposi sarcoma lytic replication. Without further ado, Rami, please.
Hello, can you hear me? All right, so good morning, everyone. Nice to see you here. Thank you for the amazing introduction, Ms. Meng. And once again, I'm Rami Al-Halak. I'll be going to the University of Pittsburgh starting this fall, and I had the privilege to be in the Hillman Cancer Center this summer under the Gao Lab. And as written up there, my project is about N6-methyladenosine, or just M6A, modification regulating Kaposi sarcoma associated herpes virus, just KSHV, and lytic replication. Whoops. All right, so let's talk about KSHV a little bit. So first of all, it's the etiological agent, which just means the precursor of Kaposi sarcoma, which causes lesions in soft tissues, primary effusion lymphoma, which is a cancer that targets the lymphatic system, multicentric Castleman's disease, which just increases risk for infections, cancers, and then most recently, Kaposi sarcoma, inflammatory cytokine syndrome, which has a high mortality rate for people who have already been infected with HIV and human herpes virus 8. So that also means that KSHV belongs to the gamma herpes virus family. But if you look at it more demographically, KSHEV was most prevalent around sub-Saharan African environments and some Eastern European regions during the height of the HIV epidemic. Now that we covered that, we can look at KSHEV in a more cellular scope. So KSHEV has two specific phases. That's latent stage and lytic activation. So during the latent stage, most cells and most genes are suppressed, except for a couple who are expressed. This includes LANA, V-cyclin, V-flip, and some microRNAs. And what they do, their job is just to overall maintain viral latency and do this by mediating the viral episome of the cell while also prolonging the cell survival and life proliferation, as much as activating the oncogenic pathways. So how does one go from latency to lytic activation? So the cell has to rely on external stimuli, maybe a stress promoter, inflammation, or whatever it may be. So it's an environmental or physiological change. So what's special about KSHEV genes is that they're expressed in a cascading fashion from early genes to late genes. So this whole process starts with an RTA, a transcriptor activator protein that just starts the whole downstream of viral gene expressions. And some notable proteins that accompany this process are ORF33, ORF67, and ORF69. They overall work together for DNA replication and viral gene expression and to produce these infectious viruses. Now that we've covered that aspect, let's talk about N6-methyladenosine, or just M6A. So just a brief history. This was first discovered in the 1970s, and back then scientists already made the link between this and mRNA instability. So it's just a sneak peek into what we're going to be talking about a little bit. So the M6A transcriptome, a transcriptome is just the overall range of mRNAs that have been expressed by an organism. They regulate mRNA fate and function, mRNA export, mRNA stability. So you can tell that there's a heavy bridge between those two factors. So how does M6A do all of this? Well, it's comprised of three parts, to be honest: writers, erasers, and readers. So what writers do, they just methylate. They add a methyl on top of an adenine or cytosine head, while erasers do the exact opposite. They demethylate, they remove that methyl head, and this includes the ALKBH5 inhibitor and the FTO inhibitor, which we'll be talking about later on in this presentation. And then finally, readers are just simply RNA binding proteins that help with pre-RNA splicing, RNA export, and RNA stability. So our research question this summer was, what is the function of M6A modification in regulating KSHEV infection and replication? And we hypothesized that these M6A erasers, FTO and ALKBH5, as previously mentioned, will increase the expression levels of KSHV, lytic replication, and enhance lytic replication by demethylating viral and cellular transcripts. So how do we go about doing all of this? We use a very, very special cell line, ISLK-BAC16 RGB. And what makes the cell line so special is that it can be examined under a fluorescent microscope. So you guessed it, RGB does stand for red, green, and blue. And it's these two different proteins that we're looking at. Red fluorescent proteins, which are going to represent latent gene expression, and green fluorescent proteins, what we're truly looking for is going to represent lytic replication. So our drugs include IOX1, C6, and C3, which will all target the ALKBH5 inhibitor, while FB232 will solely target the FTO inhibitor. And all these drugs were all under sodium Biltrate induction to guarantee results. I just put up these two images randomly for you guys to see what I was looking at day-to-day in the lab under the microscope. So I just mentioned that all these drugs will be under sodium Biltrate induction to guarantee the lytic replication, the green fluorescent proteins. But what did the cells look like without any sodium Biltrate induction? So I just randomly handpicked some drugs for you to see. So as you can see, there is no lytic replication, there's no GFP without the sodium Biltrate induction. So it shows you that sodium Biltrate induction is a very necessary factor for this process. And so moving on forward, all these images and results are at day 3 post-treatment to maximize the results. So we're studying the IOX1 drug under the ALKBH5 treatment. And for every slide I've shown 0.2% DMSO, which is supposedly our negative control to show mild results. And I showed 3 millimoles of sodium Biltrate without any drug treatment, just for comparison. So starting with IOX1, we do see there's a mild increase between 20 micromolars to 80 micromolars, but surprisingly, our DMSO treatment supposedly are negative, showed a very high stage of lytic replication. All right, but if we can examine these results under a graph, within our first biological repeat, we do see that the highest stage of lytic replication was achieved by the highest drug concentration as it should be, that being 80 micromolars at IOX1. However, strange enough, in our second repeat, 0.2% DMSO does take the lead for high stage of lytic replication, approximately 84 or 85% of GFP out of RFP. Similar enough for our C3 treatment under ALKBH5 inhibitor as well, there is a vast increase from 50 micromolars to 200 micromolars, but strangely enough, our 0.2% DMSO treatment outweighs those. And we can see these results further again in the graphs, in which in the same time, again, our heaviest dosage of the drugs, 200 micromolars of C3, does take the lead for the first repeat. And once again, strangely enough, 0.2% DMSO does take the lead for our second biological repeat. So we can guarantee this was purely on lab mistakes and mechanical mistakes on my end. And our last ALKBH5 inhibitor treatment, C6, I do want to mention some strange things. It seems as if from 10 micromolars of C6 to 100 micromolars of C6, that I like to call it even a drug overdose is what I'm hypothesizing, because unlike other treatments, this is a 10-fold increase opposing to 4-fold or by 200 times. So this is a 10-fold increase. So it seems as if the lytic application peaked around 10 micromolars of C6 is what I'm thinking right now. And of course, reluctantly, our DMSO negative control refuses to work with us. And we're looking at these graphs once again. I do want to apologize to the audience real quick. My Excel also did not want to open last night. So for the legends, it may be difficult to see because once again, my office did not want to cooperate with me. Thank you. So as our graphs, we see very rudimentary results. So in our first repeat, the highest concentration once again took the lead, approximately about 35% our GFP. And in our second repeat, it seems as if DMSO once again did take the lead for highest replication at around 85% once again. And looking at our only treatment on targeting the FDL inhibitor, FB232, again, this is pretty rudimentary results, but we do see almost as if 0.2% DMSO is showing more mild results, but this could be just, again, human technical error once again. So there's no conclusive evidence to be made, conclusive analyzations to be made from this. And we do see this information reflected once again, in which in our first repeat, 8 micromolars of FB232 takes the lead, but then slowly but surely 0.2% DMSO gains its leaderboard back at our second repeat. So what can we take away from all of this? ALKBH5 and FDO treatment may have marginal effect on KSHV lytic application, but because of how wishy-washy our results were, we have. Good morning. So because of how wishy-washy our results were, we have to conduct more repeats to make sure our results were conclusive. And we can also take other approaches such as a qRT-PCR or Western blotting, others to affirm the microscopy results. Other than that, I want to give huge acknowledgements to my PI, Dr. S.J. Gao, my mentor, Dr. Wang, for amazing guidance throughout these past 7 weeks. Of course, the members of the cancer biology head for bringing in amazing professionals in respective careers to enlarge my love for science, and overall for pushing me in the direction that I wanted. And of course, the orchestrators of this overall program, Stephen Jones and Mr. Boone. Thank you very much.
Hey, how are we doing on time? People here. Just because we're a little bit ahead, so we had family come in to decide for something. So I didn't want to. Yes. Raise your.
Hello, everyone. Good morning again. So Daniel is actually an old friend of ours. So he actually was here last summer, and during last summer, I think this kid is so talented, and also he was really excellent. So we were so happy to have him back this year. And Daniel is actually going to join Pitt as a freshman also this year, and he's going to major in molecular biology. And so at his age, I think he actually understands the projects, what he's doing on the research pretty comprehensively. And then what is amazing is that he has limited time here this summer, but he managed to get a good amount of data and which can let him draw a conclusion. And without further ado, Daniel, please.
Flip this on here so you can hold it and if you've got a pocket, stick this in the pocket.
Hello. Okay, my name is Daniel, and over the summer, I was working on a project of how metal 3 regulates KSHV lytic replication. And so KSHV, or Kaposi sarcoma associated herpes virus, majorly causes 4 different diseases: Kaposi sarcoma, multicentric Castleman disease, primary effusion lymphoma, and KSHV associated inflammatory cytokine syndrome, which are all capable of creating different malignancies. And so the breakdown of KS is that it's heavily prevalent in sub-Saharan Africa and in Mediterranean countries. And not until the 1980s during the AIDS epidemic is that the KS was prevalently shown in the United States. And so that gave people the conclusion that KSHV is an AIDS defining malignancy. And so KSHV, herpes virus, has 2 different stages, persistent latency and lytic replication. So during latency, it hides from tumor suppressors and tries to maintain cell survival. And so that is mostly done by the LANA protein, which mediates viral genome replication and persistence. And there are other proteins created like V-cyclin, K13, and B-flip, which maintain cell survival and regulate cell proliferation. And so to get from latency to lytic replication, there needs to be some type of stimuli like oxidative stress or hypoxia. And when that stimuli occurs, there is a cascade, a transcriptional cascade of 3 different genes, immediately early genes, which is mostly RTA. And what that does is it binds to its own promoter and transactivates other viral promoters. And so then the early genes, which functions in preparation of the cell ready for DNA replication and viral protein production. And then the late genes, which functions in assembling viral proteins that comprise of the viral structural components. And once after all 3 stages of genes are expressed, viral assembly begins and viral progeny is released. And in the end, the host cell is eventually lysed. And so M6A or N6 methyl adenosine is the most common RNA modification, and it composes of 3 different types of proteins: the methyltransferase writers, which compose of metal 3, which I'm working with, metal 14, and WTAP, which function in adding a methyl onto an adenosine onto a mRNA. And so what the demethylase erasers do, which compose of FTO and ALK-BH5, is they do the opposite effect of the methyltransferase writers and removing the methyl off of the adenosine. And so then the final is the binding protein readers, which have multiple functions like RNA degradation, facilitation of translation, and regulation of RNA splicing. And so we used an engineered cell line, which have 3 different proteins that under a fluorescent microscope can show 3 different types of colors: red, green, and blue. And so under the RFP or the red protein shows latency, and then the GFP or the green protein shows lytic activation. And so how the fluorescent microscope works is there's a light shined through a filter, and once it hits the specimen, a certain wavelength is shot up into a different filter, which shows the red or the green color. And so using sodium butyrate induction, we can induce mild amounts of GFP positive cells. And so I was working with 1 inhibitor of metal 3 and 4 activators of metal 3, which are denoted with the letters A through D. And so the research question I was given was, does metal 3 affect KSHV lytic replication? And based on my understanding what I did last year, I came with the hypothesis that metal-3 alters the M6A levels on KSHV transcripts to regulate their expression levels and KSHV lytic replication. And so I took 3 images per well with the RFP channel and the GFP channel. And what I did is I counted each GFP positive cell and each RFP positive cell, and I divided the GFP by the RFP to get a percent of a GFP. And so with the inhibitor STM2457, I did 3 different trials. The first 2 experiments are a repeat of each other, which shows that 200 nanomolars with STM2457 and induced with 3 millimolars of sodium butyrate show a upregulation in Lick replication. And then so in experiment 3, we increased the concentration of the inhibitor, which shows an even more drastic increase in Lick replication compared with the control. And then so the first activator A, so we also did 3 experiments. The first 2 were repeats. And what it shows is that at some type of concentration, like 1 micromolar of A, there is a decrease in Lick replication in shown in experiment 2. And experiment 1 does show with 100 nanomolars of A, there is a decrease in Lick replication. However, in experiment 3, we did a different method, and instead of simultaneously inducing the cells with the activators and the sodium butyrate, we treated the cells with the activators first and then waited 2 hours because what the activators do is it takes 2 hours for the activators to actually activate the metal-3. And so once we waited 2 hours and then induced the cell with sodium butyrate, the results show a different results than what the first 2 experiments showed, and that actually there is some increase in Lick replication, even with increasing concentration. And so with activator B, there are actually similar types of results that were shown with activator A, and that higher concentrations do show somewhat of a decrease in Lick replication. However, in experiment 3, there is some increase in Lick replication compared to the control. And so this gives us contrasting results. So for activator C, there is inconclusive results because even though the graph in experiment 1 does show 1 micromolar of C decreasing, when we increase the concentration to 10 micromolars of C, there is a drastic increase in experiment 2 in Lick replication. And then experiment 3 does show an increase in Lick replication when we waited 2 hours before inducing the cells. And then for activator D, there is also inconclusive results, as the same applies to activator C, in that with higher concentrations, there does not seem to be a decrease in Lick replication compared to the control. And that with experiment 3, like with the all other 3 activators, there does show marginal or no substantial decrease in Lick replication. And so for the conclusion, we conclude that STM2457 treatment does increase GFP positive cells in a dose-dependent manner. And so that leads us to believe that inhibition of metal-3 could enhance KSHV Lick replication, and that activators A and B inhibit KSHV Lick replication. However, activator C and D shows inconclusive results to conclude any type of upregulation or downregulation. And for future directions, we need to confirm metal-3 function with the inhibitor by QRT, PCR, and Western blotting to detect viral mRNA and viral protein expression, as well as virus titration, and confirm metal-3 function by shRNA knockdown. And so I would like to thank my PI, S.J. Gao, my mentor, Dr. Wen Meng, and the other members of the Gao lab that helped me along my journey. I would like to thank the program coordinators, David Boone and Stephen Jones, and the cancer biology site heads, Dr. Galson, Dr. Loze, and Dr. Nasser. Thank you.
Zeus, and then Anthony, when she's done, clip this on somewhere there and stick this in a pocket.
All right.
Okay.
Okay, so I hold
That's the,
this.
that's the part that is the
Oh,
microphone. This
okay.
is just the engine.
Okay.
Good morning, everyone. So it is my great pleasure to introduce our high school student, Anthony Calvi. And Anthony recently graduated from Upper St. Clair High School, and he will be attending the University of Pittsburgh at Greensburg. And he will major in the pre-med and also the minor in music. And so this is Anthony's second year attending the Human Academy. But this is the first year in Dr. Guo's lab. And it has been, over the past 7 weeks, it has been a wonderful experience for me. And I had a unique opportunity to observe Anthony's hard work and dedication in a research environment. And also he is remarkably inquisitive and always asks me the insightful questions that I explain that until he fully understands that. And he also enjoys discussing the questions assigned with other lab members, and which is pretty good. And what impressed me the most is he has a very clear career goal, and he wants to be an oncology surgeon, so which is a quite challenging profession. But he knows what he's good at it, and he is taking steps and will continue to make the efforts until he reaches his goals. And in the spare time, Anthony also has a talent, shows a talent in playing music. And he had participated in over 30 recitals, have been recognized for his excellence in playing piano. So I wish Anthony all the best as he started the college studies and also a bright future ahead. So without further ado, I will let Anthony to take the stage.
All right, can you guys hear me? All right, so thank you, Ning. So my project this summer was the structural prediction and functional validation of the 2 zinc fingers of the hepatitis B virus polymerase. So a little bit of background on hepatitis B virus, or HBV. HBV infection is a global health burden. It attacks the liver and can cause acute and chronic disease. Worldwide, it affects about 300 million people that live with chronic hepatitis B, or CHB infection. And if CHB is left untreated, it can cause cirrhosis, which is the scarring of the liver, or hepatocellular carcinoma. All right, so this is the structure of the variant. So up here, you'll notice that there's this lipid outer envelope, which contains the 3 surface proteins. There's the inner core, or the nucleocapsid, which is made of the core protein. And then inside that, you have the HBV genome, which is partially double-stranded DNA, or rcDNA, and it even comes with its own HBV polymerase right down there. Okay, so this is the HBV cycle. So when infected, here's the there's the variant up here. It binds to NTCP receptors on the outside of the hepatocyte, which then through a process called endocytosis, this then enters the cell's cytoplasm, where the nucleocapsid then becomes exposed and enters the cell's nucleus, where the partially double-stranded DNA is then repaired to a full double-stranded cccDNA. This then is transcribed to all of your RNAs, including your pregenomic RNA, your mRNAs, and others. Your pregenomic RNA then exits the cell's nucleus, and then this next part is where my project is mainly focused on. So this is boxed over here. This then is encapsulated by the core protein to reform the nucleocapsid with polymerase inside. And then polymerase carries out the reverse transcription of the pregenomic RNA to the negative strands of DNA and then positive. So in return, you started with the partially double-stranded, and then you get that an exact copy of the variant. So this is the HBV polymerase down here. And then this is it compared to the HIV, the polymerase. And you'll notice that it's fairly big in size, and it's a lot more complex. It has 4 domains, and the way you want to think about the domains is each domain has a particular part on the structure, and it also has a particular function that it carries out. So the first domain is terminal protein, or TP. It's marked by tyrosine. It marks the priming site of the polymerase and the pgRNA. The next domain is spacer, or SP. Its function is not well known. Then you have RT, reverse transcriptase, which does the reverse transcription of the pregenomic RNA. And then finally, you have RNase H, which digests the leftover pgRNA. So even though we know all this, HBV polymerase's structure is not well known. So that brings me to my next part of this, which is AlphaFold. So AlphaFold is a protein structure prediction program that Dr. Joe at the NIH partnered with the Dr. Go's lab, and they used the AlphaFold to predict the structure of polymerase. And this is what they came up with. This is the structure right here. You'll notice that, like I said, the domains, they correspond and they are color-coded to the particular part on the structure. And then you'll notice there's this highlighted bit up here. It's called the zinc finger, and this is the motif. Their model predicted that there are 2 of these located in the structure, and here's one on the left, and then there's also one here on the right, and they're made up of these 4 amino acids that meet and they bind to the zinc ion. So again, this is just another chart showing the genotypes A through H, and this is the amino acid sequences of them, and you could see that they're all lined up here, showing that in the boxes up here in green and then red, these are these 4 amino acids that make up these zinc fingers. And basically what this is just showing is that it's just highly conserved amongst these genotypes. And then you could see down here, these are the 4 amino acids that make up the zinc finger 1, and then the 4 amino acids that make up zinc finger 2. Okay, so this brings us to our hypothesis. So our hypothesis is that these 2 zinc fingers play a crucial role in the function of HBV polymerase. So to test this, this was our experimental plan. We first did a site-directed mutageneticist to mutate the amino acid cysteine 312, cysteine 341, cysteine 323, which my lab had previously done, and then cysteine 327 to alanine individually. We then transfected these into wild type and mutated HBV polymerase separately into the HepG2 cells. We isolated the HBV total RNA, the capsid pgRNA, the HBV polymerase and core proteins, and then the core DNA. And then we performed a northern blot, a western blot, and a southern blot. Okay, so the first thing we did here was our introduction of our mutations, and we did that via PCR. This is a little picture of the plasmid we were working with, and you'll notice up here, here's the HBV polymerase on it. So we introduced our mutations to this plasmid via PCR, and then after that, we wanted to see if we had product there, so we did what's called electrogeophoresis, which looks something like this. This was our result, and you'll notice that there are 3 bands going across here, just showing that our products are there. Once we were done with this step, we recircularized the DNA and moved on to our transformation. Our transformation was done by transforming our plasmids to our E. coli bacteria. And what this does is these E. coli grow on this, you see the Petri dish here, they grow on the Petri dish, and what it does is it just makes many, many copies of the plasmid so that we can work with it. So once we had our copies and it has grown in the Petri dish, we wanted to go and get them sequenced to see if the mutations were introduced. But before we do that, we have to do what's called an enzyme digestion. And then what that does is, right up here, you'll notice these 2 enzymes that are located on the plasmid. And what we do is we make a cut at these 2 points. And it separates the plasmid into 2 fragments of DNA, which then we can move forward with another electrogel,
which also shown here, which we also are visualizing the DNA to show the two separate sizes. So stuff like this right here, and then also stuff like this, that's no good. We want two distinct bands going across, showing one bigger fragment and then one smaller fragment right here. So after we got ones like this, we took them and we went on to our sequencing. So this is just a snippet of 312 and then 341. You'll notice that right here, our mutations were introduced. So this is what is going to code for the alanine. And then our 327, you'll notice that our mutation was introduced here, but you'll notice we have some missing nucleotides, which correspond to the first 4 nucleotides of the primer. So overall, we weren't able to move forward with the 327, but what we did have was we had 323, 312, and then 341. So after we did that, we did our transfection. And this is what the transfection looks like. This is a 12-well plate. We did what's called a rescue assay, where we had 2 plasmids here, one with the mutated polymerase, and then the one with, it's called HBV delta P, which basically is just the HBV genome. So, and then that process now looks something like this. Now you'll notice over here, this cycle looks familiar to the beginning, especially this part, right? This part's a little bit different because this is the transfection. But this part is similar to the cycle. And if you want to think about at the end of this part, you'd have variant down here. So that's what we're looking at. And the final part of this to wrap all of this up was we did our final 3 things, which was our northern blot, our western blot, and then our southern blot. So the first thing was our northern blot, which looks at RNA. So you'll notice up here, I have it's separated into columns. You have your wild types and then your 3 mutants. Okay, so for total RNA, that corresponds to all these parts in this cycle. So if your RNA that's in your nucleus, you have your exited pgRNA, and then you have your encapsulated pgRNA. And you'll notice that we have full expression for all of those. You have full expression in your wild type, and you also have full expression in your mutants. If we come down to our rRNA, we also have full expression there. That is your ribosome RNA. It's a control, it's a loading control to just make sure our cells are healthy and make sure the experiment is done properly. And then down here, which I think is the biggest part of the project and the big moment here for me doing this, was the encapsulated pgRNA. So you'll notice that we have under wild type, if you come down, you'll notice we have a band for under wild type, which shows our wild type is expressing this encapsulated pgRNA. You'll notice if we move over though, here we have nothing. Our mutants are not showing this step. This step is not occurring in our mutants, meaning that our mutations we introduce affect this process greatly. Okay, so moving on to our Western blot. Our Western blot looks at protein. The first box here is our flag polymerase, which is our polymerase. And you'll notice our HBC core, which is our core protein. That corresponds to these parts in the cycle. So you have your core protein. This then assembles this nucleocapsid, and then the polymerase is lumped inside. You'll notice that this part is expressed. Beta-actin is another control to show our cells are healthy through this process. But you'll notice that this process is looking good in our mutants and in our wild type. So this process was ultimately not affected by our mutations we introduced. So finally, that brings us to our southern blot. And our southern blot looks at our DNA. Our DNA corresponds to this part of the step. You have your rcDNA, and then you have your ssDNA. Now, if you come down on top here, you come down all the way down under wild type, you'll see that we have expression in our wild type, meaning that this process of the cycle is working properly. However, in our mutants, we have about 90% less yield than we did in our wild type. And this is kind of big because what this does is this just reiterates the fact that everything up until the encapsulation step worked properly, and then everything after that seemed to be hindered by this, meaning that our mutations we introduce greatly affected this step right here. So for the conclusion of this, we mutated our cysteines to our alanines, and these constructed the zinc finger motifs. The zinc finger motifs we found were greatly important to the structure of this encapsulation step. And future directions, we will create other single mutations and combinational mutations of the zinc finger motifs of HBV polymerase. And then we will investigate how these mutations affect the polymerase pgRNA interaction, the nucleocapsid formation, and then the reverse transcription. All right, so acknowledgements. I'd obviously like to thank Hillman Cancer Center Academy, Dr. David Boone, Stephen Jones. I'd like to thank my site heads, Dr. Galson, Dr. Nassar, and then Dr. Los. And then Dr. Go, I thank you for opening up your lab to me. I very appreciate it very much. And then my mentor, Ning, and then all my lab members, Chang De, Hu, Elena, Xiao, Young, and then Andrea. And then we also like to thank Dr. Zhou at the NIH for providing the polymerase model. So thank you guys, thanks.
Does anybody have a question? Thank you so much. Dr. Lucy out guarding my food.
So after Wayne is done, you want to pin this on close to you and just stick this in a pocket. Are those real pockets?
No.
I can put it on.
You can put it on there and just don't step too far away. There we go.
Hi, everyone. My name is Wayne Stoller, and my lab was the lucky lab that got to have Ginny in it this summer. Ginny Guo is a rising senior at the Shadyside Academy. Ginny actually has pretty extensive research experience for her age. She participated in the University of Chicago's Research in Biological Science program last summer, where she conducted laboratory research on the Wnt pathway. She's also working with researchers at the Beijing Institute of Hepatology, where she's performing research on new non-invasive diagnostic methods for non-alcoholic steatohepatitis. Jenny's also super involved in lots of extracurricular activities, including lots of science-related ones. She is part of the Future Health Professionals Club, the Westinghouse Science Honors Institute. She's on the debate team. She participated in the Science Olympiad. She's in the Pre-Med Club. And she's won a number of awards for these science-related activities. We loved having Ginny in the lab this summer. She was very productive, but she also integrated very well as into our team. We're really going to miss her when she leaves us. And you know, Ginny had one of the lucky situations where when we in our first test experiment, she made a very important observation that sort of changed the direction of the project she was working on. And so this is now currently the most exciting thing that's happening in the lab. And so we're super happy that Ginny showed up this summer. And I hope that you all enjoy her presentation.
Can everyone hear me? Okay. So hi everyone, my name is Ginny and I'm a rising senior from Shadyside Academy. Over the summer, I work in the Stoller lab. So today I'm going to talk about my project on investigation of therapy-induced cell cycle arrest and apoptosis in cancer. So before I delve into the details of my project, it's really important to understand what is cell cycle. So I believe many of us have learned about cell cycle in high school. Normally it has four phases, G1, S, G2, and M. But there's one very important phase that many people tend to ignore, but is especially important in cancer research, is the G0 phase, or cell cycle arrest. So the G0 phase has two types. One is the quiescence, which also refers to the reversible G0, which means that once the cells they enter this quiescence phase, they will be able to go back to the cell cycle and continue proliferating. While the other one being the senescence, or the permanent cell cycle arrest, which means that the cells, once they enter the senescence phase, they cannot go back to the cell cycle and proliferate. Therefore, theoretically, it is often considered a desired therapeutic outcome alongside cell death via apoptosis. However, what is the problem with senescence is that in recent years, a growing number of studies has shown that the senescence is actually not irreversible. So the cells, they stay dormant in the senescence phase for many, many years after the primary eradication of the cancer. And then they regain their proliferative capacity and come back, keep proliferating. And this leads us to a problem of tumor relapse, resistance to traditional cancer therapies, and therefore failures in cancer treatment. So from there, it is really important to develop some sort of combination therapy that combine the clinically available traditional anti-cancer therapy and some new therapeutic agents. So from there, this leads us to my research question: how do ovarian cancer cells choose between cell cycle arrest and apoptosis in response to therapy-induced DNA damage? So my initial hypothesis is that upon DNA damages, the cells, they remain in the G0 phase briefly with very low CDK2 activity before becoming apoptotic. So CDK2 is a protein that is the main force behind the cell cycle progression that really drives cell through it. I want to introduce one of the very important experimental approaches I use for my project, which is the use of fluorescent biosensors. So unlike the immunofluorescence many people use in their labs, where we have to fix the cells first and then stain them, with the use of fluorescent biosensors, which is coded by a DNA sequence and constructed out of amino acid, that means that those biosensors, they can be naturally expressed as part of the cell's genome, and therefore this allows us to image and quantify protein activities in living cells. One important fluorescent biosensor I use for my project is DHB. So it is essentially an indicator of CDK2 activity. When the cells, they have a low CDK2 activity, which means that they're most likely arrested, the DHB mainly diffuse in the nucleus. When the cell, they have a high CDK2 activity, which means that they are most likely proliferating, the DHB will mainly diffuse in the cytoplasm. Another biosensor I use is the 53BP1. It is a DNA damage reporter, which means that it mainly diffuses in the nucleus, and this allows me to really identify my cells and then segment them for my future analysis. So the first experiment I did is a 5-day time-lapse experiment. I used ovaricary cells. This is a model human ovarian cancer cell line. So basically, I seeded the cells, and then I treated my cells with 2 different chemotherapy drugs. The first one is cisplatin, which induces DNA damage, while the other one is olaparib, which disrupts DNA damage repair process. For each drug, I treated my cells with a high concentration and a low concentration. And then the cells, they were just happily growing in the incubator that is on the microscope, and the amazing thunder imager which will be able to take a run of images of the fluorescent biosensors every 30 minutes over the course of 5 days. So at the very end of the 5-day period, we'll be able to get a time-lapse movie for observation and analysis of the CDK2 activity. So as we mentioned earlier, we made a key discovery from our experiment is that, unlike what we initially hypothesized, the ovaricary cells, they underwent apoptosis with high CDK2 activity. So essentially, they did not enter that temporary quiescence phase before they underwent apoptosis. So I have a really cool video to show. This is in the cells I treated with a high concentration of cisplatin. So as you can see, the DHB, they remain in the cytoplasm. And then suddenly the cell, they just run it up and perform apoptosis. Similar effects were observed in the olaparib group as well. So from there, we concluded that the ovaricary cells, they did not shut down their CDK2, and they did not become arrested before initiating apoptosis. Therefore, we refined our hypothesis. We said that the cells, they essentially face this decision between cell cycle arrest and apoptosis, depending on their ability to successfully shut down CDK2 or no. So the end goal here is really to force all of the cells out of their dormant stage and into apoptosis directly. So if we want to explore this mechanism a little bit more, we use the inhibitors. So CDK2 inhibitor essentially help the cells to successfully shut down CDK2, and ideally the cells, they will all go to the senescence. Well, what is WEE1? So WEE1 is essentially an inhibitor of CDK2. So with the use of WEE1 inhibitor, we'll have a lot of cells to constantly have a really high CDK2 activity while they face DNA damage stress. So in my follow-up experiment, I treated my cells with various combinations treatments, such as cisplatin and WEE1 inhibitor combination, cisplatin and CDK2 inhibitor combination, WEE1, CDK2, and cisplatin alone, and of course control. And like the previous experiment, after 5 days, I obtained an image of myself using the Thunder imager. So for the results of this follow-up experiment, I make some graphs based on the nucleus area and the ratio of the DHB level in the nucleus and the DHB level in the cytoplasm. Based on the features of different sulfates, I divided the graph into 4 quadrants. So first, let's look at the comparison between the control and cisplatin. So as you can see over here on the control graph, there's a really large population of proliferating cells and a smaller population of quiescent cells, which is what we would normally expect because the cells, they did not receive any drug treatments. On the other hand, in the cisplatin treatment group, there is a very large population of pre-apoptotic cell present, which is also what we would normally observe because the cisplatin is a chemotherapy drug that is supposed to induce a lot of cells into their arrest or pre-apoptotic stage. What is really interesting to look at is the comparison between the cisplatin group and the combination group. So as you can see that the combination of cisplatin and WEE1 inhibitor actually decreased the population proportion of the proliferating cells by almost half, and then increase the population proportion of the pre-apoptotic cells by 4-fold, which is really good to see and kind of align with our hypotheses. So from there, we conclude that the tumor cells, they tend to choose apoptosis in a state of confusion upon competing signals of DNA damage and high CDK2 activity, which is also an indicator of cell cycle progression. So to use an analogy here is that you're trying to drive, but you have one foot on the brake while the other foot on the gas. From our follow-up experiment, we'll be able to conclude that the combination of cisplatin and adversertib, which is a drug, which is the name of the WEE1 inhibitor, can be a promising treatment that induces increased cancer cell death in a clinical scenario. For future directions, we really wanted to figure out the relationship between cell cycle phase and the cell phase. So basically, if a cell is in the S phase compared to the other cell in the G2 phase, will this affect their decision on choosing a cell phase? And we also want to obtain objective quantification of apoptosis and senescence using functional assays to confirm our results. So lastly, I want to express my special thanks to everyone in the Stoller Lab for their care and guidance. Wang, our PI, Janet, our amazing lab manager, and Dante and Betsy, my mentors, and Tyler and Rami, they are the undergraduate students. And of course, many thanks to the University of Pittsburgh, Human Cancer Center, and Human Cancer Academy for providing us this invaluable opportunity and very good experience. Dr. Galson, Dr. Lose, and Dr. Nasser, who arranged so many good lectures and talks and activities for us. And of course, Dr. Boone and Stevens, who basically coordinated the entire program. Okay, thank you.
Okay. Okay, before we break for lunch, I want to thank all our speakers. You all did a fantastic job, and it was a pleasure working with you this summer. Out in the sort of crane shed area where their food is out on a couple tables, there's some note card thingies saying cancer biology site, so you'll know it's yours. There's roast beef, turkey, and vegetarian. Vegetarians all the way in the right. So I know there's some vegetarians here, so you should go grab them. So there's drinks that you can pour, and there's stuff in the boxes besides sandwiches. Any rate, so you were sort of on your own, you can relax. We actually managed to get a photo of the kids between when we had the extra long break. I will put all our good photos together and send out some of the best ones to all the kids, and then you can share them with your families. So you all need to be back here at 2 o'clock for the academy-wide graduation and certificate. And I guess the 2-year students get some special recognition. So relax, enjoy your lunch. You can take it. There's a space downstairs that you can go to eat, or you can sit out here or wherever. Yeah, and thank you to all the parents who raised such wonderful kids. Having been a parent of 3 kids, I respect that.